Oh, sorry, I'm going to cut off this delightful banter to turn it over to Professor Ichihashi from the University of Tokyo. Please go ahead.
Okay, can I share my screen? Wait a moment. Okay, can you see my screen?
Perfect.
Okay, may I start now?
Yes, please go ahead.
Okay, thank you very much. And first of all, I'd like to thank the organizers, Irene and Nita, thank you for inviting me this workshop. I'm Norikazu Ichihashi from the University of Tokyo. Today, I'd like to talk about the evolution, Darwinian evolution of a molecular system. We used RNA and proteins, and then we performed the experimental evolution in a test tube. So I'd like to start from this picture. This is, I think, widely accepted scenario for the origin of life. I obtained from a famous review paper by Gerald Joyce. And according to this scenario, after foundation of Earth, prebiotic chemistry began, and then RNA world began. And probably before or during the RNA world, self-replication molecules should have appeared and studied Darwinian evolution. And through the Darwinian evolutionary process, self-replication molecules should have evolved, should have developed complexity to become present-day complex organisms. So we are curious what happened here during the development of complexity, how self-replication molecules allows the development of complexity. I think that this process, development of complexity, is one of the most difficult steps for the origin of life, because as I show you in later slide, in usual evolutionary experiment or molecular system, we haven't observed the development of complexity. Usually, evolution prefer simplicity. Usually, we experience the simple molecules evolved. So our research question is, how does complexity develop through evolution? I'd like to answer this question by experiment. Before starting explaining our experimental result, I'd like to introduce a possible pathway, theoretically proposed possible pathway to increase complexity of primitive replicators. Maynard Smith and Satomari proposed that diversification and complex cooperation make a complex replication network. This process studied from a simple replicator, and they assumed a replicator diversified into two different replicators which have a little different functions. And then they assumed these two replicators start to cooperate to form a large replication networks which have multiple functions, and then the RNA fused. So through this process, simple replicator became a more complex replicators. So we'd like to examine this process. This process is highly speculative, so our purpose is to examine this hypothesis by constructing an evolvable replication system and find the conditions that allows diversification and cooperation and fusion. In my talk, I show you evidences that allows diversification and cooperation, but not yet fusion. So when we started this project, we noticed that there are several self-replication systems capable of Darwinian evolution. The first such a replication system is Spiegelman's RNA replication system proposed in 1967. This is a basis for our system, so I'd like to explain in detail. The Spiegelman system consists of two types of macromolecules, RNA genome and RNA replicase, replication enzyme of RNA. Both macromolecules are originated from bacterial phage Q beta, that is RNA virus of E. coli. So Spiegelman's group purified both RNA genome and replicase and mixed and incubated in the presence of substrate NTPs. And they found that RNA are replicated by the replicase, and also mutations are introduced into the RNAs. And especially as the mutations, deletions often occurs here. And they repeated this replication for many generations through serial dilution cycles. After incubation, they diluted the aliquot of the reaction mixture with new reaction mixtures that contain NTPs and replicases. And they performed very long-term serial dilution cycle, and they found that RNA evolved to become smaller and smaller. Initial RNA is about 4,000 nucleotides, but the final RNA became 500 nucleotides. So RNA gets simpler. And also no diversity appears. The final population is almost homogeneous. So their data shows that evolution here, Darwinian evolution occurred. First, the RNA that can replicate first evolved, but evolution was not enough for diversification and complexification. So our question is, what is missing? What is missing in Spiegelman's case? So we thought that a possible answer is translation. Translation is a process to decode nucleotide sequence into amino acid sequences, and that allows a decoupling between phenotype and genotype. Phenotype here is protein, and genotype here is RNA sequences. So this decoupling, theoretically, it is proposed that this decoupling is a key process for open-ended evolution, which means that's proposed. So based on this idea, we try to introduce translation machinery into Spiegelman's RNA replication system. We called it a translation-coupled RNA replication system. And in this system, we used artificial genomic RNA. It's about 2,000 nucleotides, and it's encoded only one gene, RNA replicase gene. It is used in the Spiegelman's experiment. But here, we used different factors, cell-free translation system. This is a reconstituted translation system of E. coli. All components are purified independently. When we mix these two factors, RNA replicase is translated, and this process is different from the Spiegelman's case. In Spiegelman's case, RNA replicase was purified in advance and supplied externally. But in this case, replicase is translated from the RNA in the same reaction internally. So the following process is almost same as Spiegelman's case. RNA replicates the RNA, and mutations are also introduced. Then we repeat this process for many generations, but for evolution, we need another factor for our system, compartment. Compartment is required for our system because in this system, the phenotype and phenotype are separated into different molecules. So for selections, for efficient selection, we have to make a linkage between phenotype and genotype. Genotype here is the RNA, and phenotype here is a protein. So to make a linkage, we need the compartmentalization of the system. And as a compartment, we use the water in oil emulsion, which is a droplet. Water droplet disposed in oil phase. It is a very tiny droplet, bacterial-sized droplet. You can easily prepare such a droplet by just mixing reaction mixture in mineral oil with some detergent. And then we incubated the droplet, and inside the droplet, translation and replication occurs. And to continue the replication, we diluted the aliquot of the droplet with a new droplet containing new cell-free translation system. And then vigorously mix the droplet, and the contents of the droplet are well mixed. And then we perform the next round of translation and replications. We repeated this serial dilution cycle for many generations. And we found that when we repeated this cycle for more than 100 rounds, we found that average replication ability of the RNA population gradually increased. And also we found that mutations are gradually accumulated into the RNAs. So this data shows that RNA evolved in this case. And we also analyzed the sequence by phylogenetic analysis, and we found that the phylogenetic tree was very simple. It's almost straight. The first RNA, the initial RNA is here, and the final RNA is clustered here. So this means final RNA population were almost homogeneous. So this means that no diversity or complexity developed, no diversity developed, and also the replication scheme was almost same as even in the final RNAs. It means no complexity developed here. So we found that we introduced translation system into the Spiegelman's RNA system, but translation is not enough to allow the diversification. Then question remains, how can we develop diversity and complexity? Then we focused on another factor, parasite. We saw that parasite may play a key role. And this is because in a hypothesis of evolutionary biology, there is a red queen hypothesis. This hypothesis says coevolution with parasite may facilitate host diversification through evolutionary arms race. Based on this hypothesis, if we introduce parasitic species into our system, it may drive diversification and complexification. So we try to introduce parasite in our system. Unfortunately, we noticed that in our system, parasitic RNAs continuously appeared at higher RNA concentrations. So in a previous evolutionary experiment, we kept the RNA concentration as low as possible. Here I'll show you the trajectory of RNA concentration during the serial dilution cycle. And we adjust the dilution rate every time, so kept the RNA concentration low. But when we did not adjust the dilution rate, but at a constant dilution rate, the result was like this. Initially, RNA concentration increased because replication rate was higher than dilution rate, and increased. At this point, the replication almost stopped, and then just diluted. This was caused by the appearance of and amplification of another type of RNA. We called it as parasitic RNA. This is a small RNA, and we didn't add this small RNA in our system, but it always spontaneously appeared around 1 or 2 rounds. And this small RNA does not encode replicase gene, so it cannot replicate by itself. And in the later study, we found that this small RNA was produced from the genomic RNA by deleting internal replicase gene. So a small RNA does not encode replicase gene, so it cannot replicate by itself because it cannot produce replicase. But if this small RNA is the same compartment with other intact genomic RNA, it can be replicated using the replicase produced from the other intact RNAs. In this sense, this small RNA is a parasitic, and this original intact RNA is a host. So now we got a parasitic RNA species, and we repeat the replication for many generations. And we found that population dynamics gradually showing oscillating pattern and gradually changed as round proceeded. And we also found that various parasitic species appeared. Initial parasite is very short, 200 nucleotide, but at the midpoint, we found that very long parasite appears. And also at this point, 500 middle size of parasite appears and dominated the population in later round. We further analyzed the sequence of RNA species at this point by using a next generation sequencer using PacBioRS, and the result was summarized by phylogenetic tree. And we found that in this case, in the presence of parasites, the RNA phylogenetic tree became more complex. They are divided into several lineages. For example, here we started from a single clone ancestral RNA, and at this point they are divided into two different lineages. These lineages shown in green line we called it host lineage 1, HL1. HL1 is evolved very first, as you see.
You are at 15 minutes now.
Okay. So I quickly stop here. And this is evolved first, and the final population here contains these RNAs. And another lineage, we called it HL2, which evolved slowly using very short horizontal lines. And also another lineage is HL3, also are host lineages derived from this lineage. And from the HL2, PL2, this is a parasitic lineage, is also derived, and then accumulated mutations, and another type of parasitic lineages, PL3, also derived from here. So now we understand that RNA diversified in these conditions. Then we analyze the sequence of and the population dynamics of each lineages, and we found that initially each lineages are exclusive, but in lately they're co-replicative. For example, here I show you the frequency of each lineages, and green line HL1 dominated the population here, but HL2 were under the detection limit. But then PL2 increased, HL2 increased. Instead, HL1 almost disappeared. In this way, initially HL1 and 2 were exclusive, but in later stage, they're co-replicated together. They existed more than a certain rate. So we think that the initially HL2 and HL1 are competitive, but they find a way to co-replicate it together in the final round. To test, to understand what happened here, we chose representative RNAs at 3 time point, 3 RNA from here and 4 RNA from here, and 5 RNAs from each lineages in the final populations. And we did a series of biochemical analysis using the RNA clones and determine which RNA replicate which RNA to what extent. And then result was summarized here as a graph, and we found that initial RNA replicated itself, of course, because it is a clone. And after the division of the HL1-2, both RNA replicated almost independently, but the interaction gradually increased as round proceeded. And the final, I'd like to explain final RNA, we found that many interactions appeared between the RNAs, especially HL2. This is one of the host species, replicate itself and preferentially replicate PL2 parasite species, but HL2 also replicate all the other RNAs. It is very cooperative RNAs. In contrast, HL1, which is another host RNA, replicate itself and only one type of parasite species. So HL1 is very selfish RNAs. In this way, there is some interactions between the host and parasite species, and I'd like to, I have no time to explain detail, but we found that these RNAs interdependently replicated. If we remove one of the RNAs, the other different RNAs also remove their seemingly cooperative and interdependent. So I'd like to summarize my talk at this point. Coevolution with parasite allows diversification into distinct lineages and formation of apparently cooperative replication networks. And I think that parasite might play an important role for the emergence of life. So in the first slide, I show you a possible pathway for the development complexity, diversification and cooperation, and I show you that the similar things actually occurred in your self-replication system. So here is acknowledgement. The result I presented today is a collaboration with a member of my laboratory and previous laboratory, and supported by this funding. I'd like to thank them all, and thank you for your kind attention. So,
Hey, lovely. Just perfectly on time, and set the stage for a beautiful talk as well as staying on time. So thank you. Questions?
sorry.
People can go ahead and use the raise hand feature or put your question in the chat. While we're waiting, I have a question. Thank you for that wonderful talk. I was curious about something you mentioned very early on about how there's a hypothesis that there needs to be decoupling between phenotype and genotype in order to get open-ended evolution. I wonder if you could explain that a little bit. I'm not sure I'm not familiar with that.
Okay. His idea, Luis Minato proposed this idea. His idea is like this. Before appearance of translation, probably ribosome work as a genotype and also phenotype. Ribosome used as a template for replication and also worked as a catalyst. But this dual function may limit the possible sequence of the ribosomes. So ribosomes have to fold to achieve the catalytic function, but they have to be straight lines, unfolded during replication. So that limitation, that restriction limits a possible sequence for RNA. So that is the idea of Luis Minato. So if the function as a template and function as a catalyst was separated into two different molecules, he thinks that that separation may allow the evolution of the RNA and both RNA and protein sequences to search for larger sequence space. That is the idea.
Okay, thank you so much. Unfortunately, we're at 1:22, and we did have a question in the chat from Zach Adams, Nori, but if you can reply maybe in the chat, that would be great while we continue with our next speaker. And Irene will introduce him.
By chat.
Thank you so
very much.
All right, terrific. So our next speaker is Saidul Islam. Saidul, please go ahead and share your screen.
Yep. Can you see my screen?
Yes, we're seeing all of your slides right now.
Okay, let me just sort this out. Right, let's hide things. Blah, blah, blah.
Yep, looks good.
Okay. There's a lot of other things that I just need to close. There we go. Okay. Everything okay now? All right.
Good to go.
Yeah, good. Thank you. Yeah. So so the first session was great actually, and especially the talk of Kevin Zanli, because he partially set the stage for how the so it's partially set the stage for how the sort of primitive energetic ingredients generated by the impactors could have underwent productive organic chemistry. So what I'm going to be talking to you now is just giving you an overview of the work that we've been doing to tackle a little bit of a challenging problem, and that's how the purveyors of function, that's peptides, could have self-assembled at the origin of life. So sorry, I'm having problems. There we go. Yeah. So peptides are crucial to many biochemical processes, and so seem almost inconceivable to imagine a scenario where peptides did not play a major role in a transition from chemistry to some sort of nascent biology. And there's two major ways that biology synthesizes peptides, and one of them is obviously the one that we know very well, and that's ribosome catalyzed peptide synthesis. And this is an enormous molecular machine that translates information that has been transcribed from DNA and RNA, DNA to RNA, to make genetically coded proteins. Then we've got non-ribosomal peptide synthesis on the right-hand side, and this is non-coded, and the peptides are simpler in complexity relative to most of the proteins and peptides generated by ribosomal peptide synthesis, and it doesn't require any genetic blueprint to read off. Now, the main message I want to get across here is that the starting materials, the building blocks for the peptides, are amino acids, and they're pre-activated using ATP, and the product is a polymer. But each step is a controlled iterative coupling of an activated amino acid. It's not a spontaneous single activation event that results in the sudden appearance of a long polymer. So the question that I want to pose is, what sort of prebiotic chemistry could have come before such sophisticated complex machinery came about, but used the similar logic that biology uses today? Or the other way I could rephrase that question and pose it, I guess, a little bit more provocatively would be, does the biological peptide synthesis operate the way it does today because it's retained some of the ways that prebiotic chemistry operated back then very effectively at the origin of life, before enzymes were even on the scene? So it's difficult not to talk about amino acids and peptides without referring to the Miller-Urey experiment. And what this first demonstrated was the detection of amino acids under simulated pre-biological conditions by providing a lightning discharge through a mixture of gases that led to the detection of some of the proteogenic amino acids. On top of all this, though, there's a lot of other stuff going on there, because you can see from this picture there's a formation of a tar. And this tar is caused by runaway reactivity, presumably because the conditions for these reactions just a little too aggressive for the first phase of productive and selective organic chemistry to generate materials that could assemble into the first biopolymers. But it's not the only way that organic material could have been generated from inorganic substances on the planet. There's probably other ways that we could do it. So I don't want you to concentrate on this slide too much, but what I want you to see is that recently chemists, and this is work from John Sutherland's lab in Cambridge, and including us, have been thinking about the problems of prebiotic chemistry from a systems chemistry approach. And this is leading to the discovery of cleaner, more efficient, selective and robust prebiotic chemistries under very mild conditions. Now, the systems chemistry approach considers chemical ensembles and their sort of mutual reactivities in mixtures rather than in isolation. And what it does is it harnesses the abilities of apparent byproducts to feed into other productive abiotic processes. And so what we see here is a network of reactions that's been termed cyanosulfidic protometabolism. And it's a purely chemical system that evolves from cyanide as the inorganic feedstock alternative to carbon dioxide chemistry. Now, hydrogen cyanide undergoes reduction in the presence of UV light to generate formaldehyde and ammonia, and that reduced carbon and nitrogen is generated in one single step. And the reaction network, when fleshed out, generates a library of RNA, lipid, and a dozen of the 20 biological amino acid precursors. Now, these precursors are aminonitriles, and I'm going to be concentrating on that for the rest of my talk. So the prevailing assumption has always been that aminonitriles that were generated by cyanosulfidic protometabolism are the precursors of amino acids, but they can only become amino acids after they've undergone hydrolysis by water. But there are problems with this, because if you hydrolyze to hydrolyze nitriles with water, it's a very slow process, and you usually need very strong acidic conditions. And that also would destroy any peptides that were present as well. But the hydrolysis also does something else, because it's really detrimental, because what you're doing is you're losing the inbuilt reactivity of the nitrile that comes directly from its generation from hydrogen cyanide chemistry. And so then what we need to do after that is to provide energy into the system, either in the form of heat or chemical energy, or we have to try and promote peptide synthesis over competitive hydrolysis by dry state heating to turn them into peptides, which end up being polymerization reactions. Now, the thing about uncontrolled polymerization chemistry, whilst it's conceptually simple, it can in practice be quite inefficient, because chemical activation is typically incompatible with a lot of the amino acid side chains, and this gives rise to other potentially unproductive side reactions. So perhaps we should start looking at converting the aminonitriles directly to peptides and bypass amino acids altogether. And this proposal seemed quite appealing to us at the time, because it uses the natural reactivity of the aminonitrile rather than dissipating the energy. So our solution to the synthesis of peptides came from clues about biological peptide synthesis that might bear some relevance in ancient prebiotic processes. And what you can see here is, in biology, N-acylation of peptides is more prevalent than one might realize, because it's certainly something that I didn't really know at the time, and I think it's something that's still poorly understood. So N-acylation occurs in ribosomal and non-ribosomal peptide synthesis, and it could be because it is stabilizing the peptide that's forming, because degradation of peptides during chemical synthesis usually occurs because of backbiting and truncation of the N-terminus of a peptide. Now, what's really interesting here is that there are two forms of ribosomal peptide acylation. There's N-formylmethionine, which is the starting residue of coded peptide synthesis in bacteria, and then there's N-acetylation, which is a post-translational modification that occurs after the peptide exits the ribosome. And in fact, about 80% of biological peptides are N-acylated. So what we did was we used these lessons from biology, and we arrived at the nuts and bolts of converting an aminonitrile to a peptide. And this chemistry bypasses amino acids altogether. So what we found was we can directly convert aminonitriles to peptides by doing this initiation by N-acetylation. And what this does is it lets us harness the inbuilt energy of the nitrile, and all of this can be carried out in water, and there's no need to overcome the barriers of amino acid couplings that would be faced in water, because hydrolysis is no longer a problem. And in fact, in this chemistry, I'll show you that water is necessary. So what we find is once the acetylation has taken place, it activates the nitrile towards the addition of hydrogen sulfide. And hydrogen sulfide generates this thioamide, and then this is primed for hydrolysis by the addition of water to generate what is a stable thioacid. And then this, in the presence of iron cyanide salts, undergoes activation and then coupling with another aminonitrile. And so what you've done is you've generated a peptide bond in water without using an amino acid. So then we did this, so iterated this process, and we ended up with, forming oligopeptides in really, really good yield. And what we also found was that all the amino acid side chains can be tolerated by coupling every single amino acid, every proteogenic amino acid, and we found that it could work in really high yield with high generality. So that's quite a tidy solution to generating peptides in water, and it allows us to bypass amino acids, but can we improve upon it? So we moved on to something at the time felt quite very challenging, and that is catalytic peptide synthesis in water. And this is an area of research that not only interests us as prebiotic chemists, because catalysis by organic molecules in water are rare, but it's also of interest for those working in synthetic chemistry and catalysis, because making a peptide bond in water, which in itself is vulnerable to hydrolysis in water, is really, really challenging. So can we catalyze a peptide synthesis in water that doesn't catalyze the addition of water itself? So we return to this slide again, and we can again learn a few things from biology. And this is the non-ribosomal peptide synthesis cartoon that I showed earlier on. And what's interesting in this slide from an origins of life perspective is because there are hypotheses that thioester-mediated chemistry may have enabled anabolic processes at the origin of life. And these proposals have a little bit of merit, and this is why, because if I strip away this chemistry, all this complexity, and just draw a cartoon, what you're left with is a very simple process where an activated amino acid, which has been activated with ATP, is charged onto phosphopantothene, which is a thiol, and then this thioester undergoes iterative peptide bond formations. Now, the role of sulfur of pantothene cannot go unnoticed, and it's essentially a key catalytic residue, but it still needs activation of the amino acid with ATP. So if we want to use amino acids in catalytic peptide synthesis, we need a selective prebiotic activating agent in water. But sulfur and nitriles, as I've shown you in my earlier slides, have a unique affinity for each other in water. And so what we found was we found a very special and rare example of organic catalysis in water, organocatalysis. And what we found was peptide ligation is catalyzed by various organic thiols that allows a direct coupling of peptides with peptide nitriles with no additional activating agents required anymore. And this process is incredibly simple because all you need to do is one just incubates the three components together and a peptide product emerges. And it's just as simple as that. And what's nice about this chemistry is it works at pH 7. It tolerates all the amino acid side chains again without any protecting groups. And the only byproduct is ammonia. And ammonia is a useful byproduct in a systems chemistry scenario because it can then be returned back to generate more amino nitriles through the Strecker reaction. So what it does is it raises an interesting question of whether this sort of chemistry might remain imprinted in some of the way, or some way in biochemistry today. Because if we compare the core basics of our catalytic peptide ligation cycle and non-ribosomal peptide synthesis, we can see quite stark similarities. So the difference between the nitrile, the difference being here is that the nitrile is coming in already pre-activated. There's no need for ATP or an ATP surrogate. And the substrate that comes in with the activation energy installed, the nitrile, is an excellent prebiotic candidate as a potential primordial energy currency, and which may have come before, I suppose, ATP or thioesters. Now, in this catalytic peptide ligation, we discovered many water-soluble thiols as catalysts, including coenzyme A and various cysteines and glutathione. But cysteine serves as a precursor for sulfur-containing biomolecules like coenzyme A, and cysteine is a major pathway for incorporating inorganic sulfur, so like hydrosulfide, into biology, and it's critical in numerous biological processes. So how does biology generate cysteine, and can it inform us in a way for prebiotic chemistry and how it could have produced it? Because generating cysteine prebiotically is very challenging because it's a very reactive amino acid side chain. So biology makes cysteine in a really odd way. So if you considering most of the incredible things that enzymes can do. And so what it does is it swaps the oxygen of the amino acid serine for sulfur in a multi-step pathway. What it does is it first acetylates the alcohols of the serine side chain whilst it complex to pyridoxal phosphate. And then what that does is it lowers the energy barrier to eliminate acetic acid to form dehydroalanine, this DHA species here. And then that is intercepted by hydrogen sulfide to generate cysteine. And what's astonishing about it is that biology has to resort to these such steps to force through such a mundane chemical transformation as simple as dehydration, because enzymes can routinely do this. But in this case, acetic acid is a better leaving group and a stronger acid than water. But it begs the question why it has to do this. Could it have been because it's a leftover from a bygone prebiotic chemistry? We don't know, but it might be because of the sort of the chemistry that the chemistry might be an imprint from nitrile chemistry that follows the same logic. So what we did and what we found was we could copy this biology using nitrile chemistry again. So in our case, we take serine nitrile, and we can it undergoes complete acetylation in water in one step. And what's remarkable about this reaction is that we're acetylating an alcohol in 55 molar water. But it's the formation of the dehydroalanine residue at neutral pH that really took us by surprise, because the chemistry is all nitrile activated. And if you try and do this chemistry with the amino acid, it just doesn't work at all. So this dehydroalanine nitrile that we produce can then be intercepted by hydrogen sulfide or another sulfur surrogate thioacetate, and then this can be go on to become various cysteine derivatives. But what's interesting is that we've generated this cysteine residue in a catalytic form, and this single amino acid residue is a rudimentary enzyme to carry out the peptide ligations that I showed you in the previous slide.
5-minute warning.
Thank you. Yeah. I think I went quite quickly there, so I'll just summarize. So just to summarize, the general theme that ran through my talk was that we're trying to harness the natural reactivity of nitriles that selectively assemble to generate biomolecules. And this all had to happen without the assistance of, I suppose, the mechanistic wonders of biological enzymes that would otherwise offer specificities and selectivities that we can only dream of in prebiotic environments. And what we can see from this chemistry is we can see the emergence of function from small molecules generated from the abiotic chemistry of hydrogen cyanide. Now, in a prebiotic setting, we require a buildup of a lot of material for an emerging biology to feed on. And so once rudimentary catalysis is in place, which we've already seen with cysteine, for example, we can start imagining more complex catalysis of difficult reactions starting to emerge, mainly those ones that we see operating in present biology today. And so what was needed on an early Earth is a lot of productive organic synthesis to outcompete degradation processes, and the cyanide chemistry that I've sort of touched upon in this sort of whirlwind of slides would profitably fulfill that role. So I think I'll leave that acknowledgement slide there, and I think I went a bit too quick there because I was a bit concerned about going over. So I'll take as many questions as everyone would like to sort of give me. Thank you very much.
Thank you, Saidul. Very, very nice. So the floor is open for questions. Put them in the chat or raise your hand. While people are typing and gathering their thoughts. Oh, okay. I see one in the chat from Felix Mueller. Great talk. Can you give us an idea on the half-life time of such aminonitriles towards hydrolysis in water, maybe even at different pH values?
So I haven't specifically looked at that chemistry, but the hydrolysis of aminonitriles, I guess it depends on the conditions that you're doing it at, and I guess and the pH and temperatures. But a lot of work has been done on the sort of the hydrolysis of aminonitriles because of its industrial value, and a lot of work was done by the group of Cameras and Robert Pascal. And one of the things about nitrile hydrolysis is a lot of the chemistry that's been sort of attempts to catalyze the hydrolysis is looked at using aldehyde catalysis. And what it does is usually you need to use very high pH to hydrolyze it to an amino amide first. And then after that, the aldehyde catalysis doesn't really catalyze the hydrolysis to the amino acid. So in the chemistry that I've presented here, we don't detect hydrolysis of the aminonitriles because the chemistry, I suppose, under sort of hydrosulfide conditions is much faster than the addition of water that can take place. I hope that answered your question, Felix.
Yes, very nicely. Thank you.
No worries.
Okay, that's perfect timing. I hope that you'll be able to stick around for the discussion session, Saidul, so we can talk more. I want to ask you some things too.
Yeah.
It's our next speaker is Wilhelm Huck. Are you there, Wilhelm?
I am indeed. Yep.
Okay,
I'm ready
terrific.
to share my screen. Yeah.
Okay.
Very good. We should be. Okay. I hope it's full screen now.
Looks good. Thank you.
Okay. Thank you. Well, let me thank the previous speaker in a way for a very nice introduction. I can take it from there in a way, yeah, because I wanted to take you from sort of the synthesis of molecules to the the synthesis of cells, the origin of cells and the origin of evolution. And so what the previous speaker has shown, I believe, is that we chemists, other chemists that are better in synthesis than me, have found many different prebiotically plausible pathways to pretty much all the building blocks of life. And if we assume for a moment that indeed on prebiotic Earth, all the molecules that life needs were present, then do we really have a living system? And I think the answer we all appreciate is that that is probably a no. Because in the end, it is only life when everything comes together into some kind of first cell where there's a membrane, there's a genome, there are enzymes, there's a metabolism, and all of that essentially needs to come together. And so I think we probably understand how to synthesize these things, but all of this at the same time, we don't really know where that comes from. So to get to this first living system, we need to have some kind of evolution. And the evolution I'm talking about is not evolution as in Darwinian sense of evolution, but some kind of prebiotic evolution that took us from this collection of molecules that we now have to something that we can probably recognize as alive. And we probably don't know how that looks like yet, but there needs to be some kind of process. And that is the question that I want to discuss today, is what is that process that actually drives the obvious appearance of these molecules towards something more complex, towards a system that then eventually crosses some threshold and then it becomes alive. And then apparently this must be a process that is so powerful that once it happened some 3.8 billion years ago, it hasn't really never gone away. So it might have started with some small bumps, but once it was really alive, I guess it has been alive ever since. And so this is something that we don't really understand. We don't know the driving force for this prebiotic evolution. So we don't really know what to look for. And what I was thinking is that you could say, well, evolution in a very general sense is that life adapts to changes in the environment. And I was thinking, can we find something, a chemical equivalent, where reactions adapt to the environment? And then I don't want to study a single reaction. We need to study a system. And the system that I want to study is the so-called foremost reaction network. And so the foremost reaction in principle is very simple. You start with just two molecules, formaldehyde and dihydroxyacetone. And if you cook this in the presence of calcium and sodium hydroxide, let's say pH 12, 13, then you get, after a few moments, you start to smell caramel. And if you wait a bit longer, then it becomes black tar. So for a long time, people thought, this is an intractable mixture. But actually, if you look at it, it's not so intractable. It might generate a lot of different compounds, but the underlying algorithm, if you wish, is actually rather simple. So there are aldol addition reactions, and there can be aldol addition reactions between the formaldehyde and some higher sugar, or between different sugars that are already 2 carbons or 3 carbons, or indeed 4 or 5 carbons. The aldol addition is reversible, so we can also draw retroaldol reactions. And in the end, you have proton transfer. You're working on the basic conditions. And there is a bit of Cannizzaro reaction, which is a disproportionation between aldehyde and a ketone towards carboxylase and and alcohols. So small number of reaction, enormous number of compounds. And so this is a system we wanted to study oh, by the way, you can take any starting sugar. So you can take dihydroxyacetone, I can take any sugar, and you will still be able to generate all the different molecules in your network. We also truncated the drawing here because it keeps going and going and going the longer you leave the reaction. So the way we study the reaction is by not actually running it in a flask and running it to black tar, but running it in a so-called flow reactor. And if you do this in a flow reactor, then essentially the average residence time determines the reaction time of your reaction. And so in this case, we flow in formaldehyde, calcium, hydroxide, any of your starting sugars, and then you collect the outlet of your reactor, and then you derivatize it. There's a nice method from Oliver Trapp in such a way with silyl ethers that I can actually see it on a GCMS. So if you then inject it into a GC, we have standards to sort of tell us the intensity of these peaks as well. Then we can sort of measure for each experiment how many C6 sugars, C5 sugars, C4 sugars, C3 sugars were formed, and how many of each. So we quantitatively follow about 51 different sugars that are formed, and that is approximately 70% of the mass that we can recover. Of the remaining 30%, I think most of it is formaldehyde that actually evaporates during our workup. And then we test lots of different reaction conditions. As I said, we were interested in finding out how this reaction adapts to the environment. And then we collect lots and lots and lots of data. What we then thought is that, well, for each reaction, so each bar chart here is one experiment at a different condition, where you would say, well, if I added more formaldehyde or more calcium, or change the pH or a different sugar, different reaction time, different temperature, I see different C6 sugars, C5, C4, C3 sugars, and I can see maybe trends. Well, after a while, you can convince yourself that you see all kinds of trends, but in the end, there's no trend with sort of changing a parameter in the environment to changing the composition of your mixture. So after a while, you have to resort to other techniques to analyze what is going on. And in our case, we use a clustering method that will sort of group compositions that are most alike into some kind of hierarchical structure. And so in this case, we choose to depict this as a dendrogram, which looks like this. So the leaves of the dendrogram are your bar charts that I showed you before. These pie charts are essentially the composition of my mixture. And every time we have a branch, then everything in this branch is more alike to everything else in that branch than to any of the other branches. But of course, if you're looking inside the branch, then you have side branches that are again more alike than others. And so now you think, okay, now we can try and understand what is going on in the foremost reaction if we look at the composition on these different branches. Actually, that is not the case. So if you take one of these examples where we change the formaldehyde concentration to ever higher concentrations, and concentration of compounds go up, some go down, some stay the same. If you look in your dendrogram, then you start with 5 millimolar, 10 millimolar, 50 millimolar, and then it goes up and up and up to 200 or 400 millimolar, and it's all over the place. It's a different branch. Then you do the same thing for calcium and hydroxide. And again, it goes from one branch to another branch to another branch, which simply means that this reaction is doing more than just changing the composition. It's all over the place. So if you then say, well, how do we find out what is really going on? We thought we have to actually look at the reactions that are taking place. So in this case, we first give no longer a constant input, but a varying sinusoidal input of dihydroxyacetone. And then we look at the formation of lots of different products in our network. And of course, they sort of oscillate with your input, but they all have a bit of a phase shift and they have a different amplitude because they might be further away from the origin of that oscillation. And so if you then teach the computer chemistry, then the computer can tell you that if you start with dihydroxyacetone as an amplitude and you get different compounds with a certain amplitude, the computer can tell you which kind of reactions should have taken place to get you to that particular reaction. And if you then see what happens at 5 millimolar, 20, 100, 200 millimolar, you see that this complexity of the network increases. More and more reactions are taking place that tell you, that explain to you how you get to the particular composition that you find for each of these different reaction conditions. And so instead of looking at the different branches by looking at different compositions, what you should do is you should look at the different branches in terms of a heat map of all the different reactions that took place. So what we've done here is we have made a very detailed list of all the possible reactions at the different carbon units, and then you count how often they occurred based on the composition of your reaction. So they must have occurred. If you start with formaldehyde and dihydroxyacetone, these reactions must have taken place to account for the composition of the mixture. And now what you see is that the reaction essentially self-organizes. These different branches, you say sub-branches, they tell you sort of reactions that, or networks that are more alike to one another, therefore producing the same output. Now you can zoom in, and I think in the light of time, I don't want to go into too much detail, but you can see two examples here. Some of the branches are actually very simple. They are essentially just dihydroxyacetone condensations, and they occur at very, very low formaldehyde concentrations, which kind of makes sense. If you add more formaldehyde, then you get more aldol-type condensation reactions, and they are sort of driving C1 carbon chain extensions. If you add everything together, then indeed you can start to find the Breslow cycle that everybody is familiar with in the foremost reaction. But this Breslow cycle is not the foremost reaction. It is essentially the central point where different pathways come together. And from here, essentially the sugars are fed in, and they're going in different directions depending on different reaction conditions. So dominating either towards condensation reactions, aldol reactions, etc. So what is another way of looking at this? Well, the way I look at it is maybe from this children's toy here. If you start pushing this children's toy, then it will sort of adopt all kinds of different shapes. But if you then let it go again, it will return to its original state. And so in our case, the actual sort of colored dots here, they are your molecules. And the black strings, they are the reactions connecting these molecules. And so when you push on this network that we have, then it rearranges the balance between these different reactions. And that is why the overall composition will eventually be different. Now, what we have done so far is essentially we studied the output of the foremost reaction under some kind of steady state input concentration. But life obviously has different environmental conditions that actually change all the time. So you could say if I have some kind of noisy input of calcium hydroxide, then I will also get some noisy output of all the different compounds. And so what we do in our next experiment, we are not going to sort of keep it at any one of these concentrations, but we just give a series of, well, at 30, 40 more minutes of varying input concentrations, and we keep collecting the output, and we then sum it all together to see how this noise actually propagates into noise of concentrations in the network. And so when you do that, take you through this slide over here, is these were all the steady state concentrations. So if we just have a fixed concentration of calcium hydroxide, and I run it at 20 millimolar, 50 millimolar, or 100 millimolar, then over time, if I collect 50 to 100 samples, there is a bit of variability in the concentration of compounds formed. This is dihydroxyacetone, this is C4 sugar, erythrulose, C5 sugar. Then you see there's a certain distribution. That's fine. It can be sort of multiple steady state in the network. It could be different explanations. But what you see now is if I give a calcium hydroxide sort of noise with low or high amplitude, I get a different overall average value for the concentration of dihydroxyacetone in my mixture. And this is the same for every network we study and for every compound that we study. So to explain it in another way, if you normally say, if I have a reaction and I add more or less catalyst, and I do this in a random fashion, and I keep measuring the output over time, then on average, I'm going to produce exactly the same amount of compound, simply because the reaction sometimes goes a bit faster and sometimes a little bit slower. So on average, it goes the same speed. What you see here is that is not the case in our reaction network. If you have a high amplitude noise or a low amplitude noise, your average composition in the mixture changes. And it changes in all kinds of directions. So in this case, you can see here these compounds change in different directions. We take more compounds, and again, they go up or down, or they stay more or less the same, but become a really broad distribution. Anything sort of can happen. And so this is a really strange thing, because this means that the foremost reaction network not only adapts to a different environment, but actually also has a temporal component in there. So it keeps track of changes in time. And so one way of looking at, well, you can do different frequencies, and again, it also responds to different frequencies of a change in the environment. So you can sort of do this in a very extensive fashion. You can also say, well, I give one input signal that is composed of 3 different time series of 30 seconds, 60 seconds, and 120 seconds periodicity. And then I keep measuring the average output over time. And what you then see is your compounds start to cluster. So the compounds cluster, and they have different correlations with different frequencies of your input signal. And so what is happening here is that your central Breslow cycle, this is the central cycle where all your molecules were coming from. But there has to be a certain reaction that actually connects the central Breslow cycle to the different molecules in the different clusters that we find. And what we believe is that these gating reactions, they're responsible for filtering out different temporal fluctuations in the environment, thereby steering the network overall in one direction or the other direction, depending on the amplitude or the frequency of the temporal variations in the environment. So let me try and finish this up. Why is this important? I think, well, life originated in a dynamic environment. And I think what we see is that the dynamic traits of these environments, they shape the outcome of the networks that we form, and also the outcome of the chemical composition of the prebiotic soup. And so they might have selected for certain chemical reactions that were in tune with the changes in the environment. And so you could also say, well, what my foremost reaction reflects is some kind of sort of processing the information of the temporal variations in the environment. So maybe what life was doing all along, the driving force for this evolution, is the processing of information. Don't know how to quantify that yet. A general message is I think we have to move beyond molecules and start studying the properties of these systems. I think also we have reached the end of what we can do, I think, by just manual experimentation. I think we have to now, if we want to go forward, we have to combine high throughput experimentation and probably some kind of machine learning to tell us what is going on. And I think if I have more time, I can tell you what you can do in terms of unconventional computing using these chemical reaction networks. However, I think time is probably running out. So let me thank Will, Lena, Peer, and Thijs, who have been doing all this experimental work and done all the analysis. Lots and lots of GC spectra, lots and lots of different ways of looking at the data. And thank the Simons Foundation for funding this work. Thank you very much.
All right, thank you so much, Wilhelm. This was perfectly on time and lovely presentation. So questions, there's a few questions in the chat. Let's see. Omer Givon says the usage of GCMS could help you understand the system from the pH point of view. I don't know if you want to respond to that.
I didn't quite understand that one. Well, I mean, if we vary the pH, so we change the sodium hydroxide concentration, then we do see that the composition of the network changes quite a lot. I must also say is that we have only explored the very high pH regime. So we haven't really gone to, well, we have done some experiments at pH 7 and 8, but not really extensively. So I don't really know how the network looks like under those conditions.
Okay, good. We have 2 or 3 more questions. So very quickly, Juan Perez Mercader says, very nice. What are the statistical properties, that is the probability distributions of the noise? Are they correlated to their averages?
Yeah, so difficult to say because we put in a Gaussian noise profile. So you can calculate that and in your pumps, can sort of ensure that that is the input that you give, but we don't take samples for that long that we really sort of know that the noise completely reflects the input variability that we put in, or whether it's skewed towards certain frequencies. I don't know yet. So it's a good point. We haven't completely analyzed that.
These are all great questions. And folks, remember that there's a longer discussion section after. So yeah. All right. And then Stuart Bartlett asks, is it possible that these kinds of reaction networks are manifesting some kind of Hebbian learning?
Yes.
Hebbian.
So it's not Hebbian learning, I think, in our case, but what I can show you is that these things can do reservoir computing.
Okay. And finally, Saidul, you have a question. Go ahead, please.
Yeah, well, Wilhelm, that was a really nice talk. I just wanted to ask you, did you try and because we know that borate can sometimes constrain the formose reaction. So I can't remember the details of your paper. I looked at it very quickly once, but did you see any sort of selectivity, or did you see a reduction in complexity when you use borate?
No, if you use borate, then everything stops. So essentially you stop the complexity completely. Which is not what we wanted. So that's why we didn't do that. What we hope is that ultimately, if you have machine learning and high throughput experiments, maybe your machine learning will allow you to push it towards, say, the selective formation of ribose or the enriched formation of ribose. We do find ribose sometimes, but in very low concentrations, actually. So the borate will allow you to do that, but then it shrinks the network so much that it is sort of almost a straight line of reactions.
Okay, I'll ask you more later on if you're around. Actually,
I might be, but I'm not 100% sure.
Oh, it's quite late for you. Yeah, sorry.
Yes, it's late.
Wilhelm, I have a question, but I'll save it for another time.
Another approach, which is very important, is to investigate, for example, extraterrestrial matter, such as meteoroids. For example, in 2014, the Rosetta mission brought a mass spectrometer to a meteoroid, and small molecules composed of carbon, oxygen, nitrogen, and hydrogen, such as aldehydes and nitriles, could be identified. Molecules that could have been the starting point for chemical evolution. Since chemical evolution had billions of years of time, and PhD students instead have rather limited time for the experiments, how can these hints from atmosphere chemistry or from these space expeditions be used to create routes towards complex biological molecules such as RNA? It is generally agreed on that RNA must have played a crucial role in the origin of life due to its position in the central dogma of molecular biology connecting genotype and phenotype. But RNA is not just carrier of genetic information, but can also act as a catalyst itself, as it was shown by Thomas Cech and Sidney Altman, thereby making it a very promising candidate for the explanation of the origin of life. So how can the monomer building blocks, the nucleosides, be formed from initial building blocks such as ribose or a nucleobase? It is not as straightforward as one could imagine that you can perform a direct glycosylation of both of these compounds, ribose and a nucleobase, and thereby form the desired nucleoside. It was shown by Orgel in the 1970s that when you approach such a reaction, it was found to be unfavored, the formation. Instead of getting the desired N9 adenosine product, the side product N6 adenosine was the major product that was detected in this experiment. Not just for the purine glycosylation, but also for the pyrimidine glycosylation, it shows difficulties. The direct glycosylation of pyrimidine nucleobases applying the same reaction conditions as used before showed that no product was formed either, which is explained by the lone pair delocalization of N1, both for uracil and also for cytosine. So alternative pathways were required for the general formation of pyrimidine and purine nucleosides. The pioneering work on this indirect glycosylation pathways was demonstrated by Leslie Orgel, who used ribose and cyanamide and also cyanoacetylene to form the nucleobase stepwise on the ribose. Such type of de novo formation of the nucleobase from cyanamide and cyanoacetylene was also later adopted and further investigated by Matthew Pauner and John Sutherland in 2009. In the blue pathway here, we can again see the direct glycosylation approach, which turned out to be not functional. So taking the ribose and the nucleobase, which did not lead to product formation. But in the green pathway instead of starting from ribose, more simple precursors, such as glycolaldehyde or glyceraldehyde, were used. After the reaction of cyanamide with glycolaldehyde, the 2-amino-oxazole was obtained, which after reaction with racemic glyceraldehyde gave a variety of different stereoisomers, of which the arabinose oxazolene can further react with cyanoacetylene to form the N-hydroarabinonucleoside. And then in a final phosphorylation step, the 2'3' cytidine cyclophosphate could be obtained in this study. As seen from the previous studies, it remained difficult to control both regioselectivity and stereoselectivity. So 6 years ago, the Carrel group proposed a pathway towards purine nucleosides, as you can see here in the red highlighted region. This pathway was based on the findings of Eschenmoser. Eschenmoser showed that glycosidation of form amidopyrimidines, here number 26, with ribose and subsequent ring closure leads to the formation of purines. Thereby, the regioselectivity problem could be overcome due to the C2 symmetry of the form amidopyrimidine. So both this amino group, but also this amino group, lead to the right formation with the right regioselectivity. And this form amidopyrimidine can be obtained from the corresponding nitrosopyrimidine, as it was showed by the Carrel group, in a reaction with formic acid. Then, additionally, 3 years later, a new synthesis towards pyrimidine nucleosides was conceived in the Carrel group, starting from cyanoacetylene and hydroxylamine, yielding 3-amino isoxazole, as you can see here in the upper part. This isoxazole can then be reacted with urea, forming the urea isoxazole, which then can be glycosidated with ribose. And a final reductive opening of the isoxazole and intramolecular cyclization leads to the formation of the pyrimidine building blocks, thereby creating 2 roots from the Carrel group that can form purines and also pyrimidines under prebiotic conditions. Of course, the formation of nucleosides, as it was already clear from previous prebiotic research, is unfortunately not a straight arrow, as it was indicated by the previous unifying scheme, especially when it is about the investigation if these 2 routes can be performed simultaneously. So in our geochemical model, we assume that the chemistry took place in several basins to locally separate reactants and intermediates. Then in wet and dry cycles, they can tune the concentration and thereby promote chemical reactions of the molecules. To separate insoluble materials such as the nitrosopyrimidine, it is also needed to have several basins, and that soluble materials can be separated from the precipitated compounds. Then in a later stage, these separated molecules can afterwards be combined again after undergoing another reaction, and then in the dry down experiment form the nucleoside. The ones highlighted here in red in the chromatogram of this final one-pot reaction, they are referred to the purine nucleosides, and the blue signals here in the chromatogram refer to the pyrimidine ones. As it is often the case in prebiotic chemistry, various side products can form, as we saw before, either stereoisomers, regioisomers. Solving one problem is not necessarily solving every problem, then another problem arises potentially. For the example of the adenosine formation from ribose and from amidopyrimidine, 4 different reaction products could be observed. Both furanoses and pyranoses were formed along with their alpha and beta anomers, and the canonical beta furanose was formed here in 20% yield. Now showing these rather complex chromatograms and mixtures of various building blocks that are typical for prebiotic chemistry, I would like to move over to the second part of the presentation with a question. So is it really plausible that life exclusively originated from an RNA world composed of the 4 canonical nucleosides, or could there be a bit of a coevolution of RNA and other RNA molecules, or even of RNA and peptides? It is mainly agreed that RNA must have played a key role in the origin of life, but as we have seen already from the Urey-Miller experiment, the formation of amino acids was also observed under prebiotic conditions. So especially when we think about the biological translational process, is it plausible that such a complex machinery made of peptides and RNA has evolved in a single step, or was it a stepwise evolution from RNA and amino acids? So having a closer look into the contemporary peptide synthesis could give us some further hints on how RNA and amino acids could have co-evolved. One of the most central molecules in the translational process is the tRNA. Here you can see the famous cloverleaf structure of the tRNA, which was first elucidated by Robert Hawley in 1965, who later on was also awarded with a Nobel Prize for his findings. The tRNA functions as the carrier of the amino acid, which is connected here to the 3' terminus. But looking here at the structure, it's already clear that the tRNA is not as plain as one could imagine. So you can observe here a lot of modifications, for example here pseudouridine, methylinosine, and many more. Then over the past decades, of course, additional modifications were found in the tRNA. We are especially interested in the modifications here very close to the anticodon loop, for example here in position 37, where we can find amino acids as well. So the amino acids are not just attached to the 3' terminus. Of course, that's the spot where the transfer of the amino acid happens. But in the tRNA, as post-transcriptional modifications, there can be also amino acids found as modified nucleotides. For example, here this T6A modification. In very close proximity in certain tRNAs, there are also some aliphatic amino groups. For example, here there's methylaminomethyluridine. So seeing these 2 modifications, we were wondering, is it actually possible to have a simple and primitive peptide synthesis based on the nucleobase? So far, all of the approaches towards peptide synthesis in prebiotic context rely on either ester bonds to the ribose or certain modifications that are attached to the 5' position, to the 5' phosphates, either via this mixed anhydride or phosphoramidates. So, additionally, it has been shown that these modifications are not just biologically abundant, but can also be formed under prebiotic conditions. As it was shown by Stanley Miller in 1995, he was investigating the formation of the methylaminomethyluridine. They could show that starting from 5-hydroxymethyluridine upon a nucleophilic substitution, indeed the methylaminomethyluridine can form. In 2018, it was shown by the Carrel lab by Christina Schneider that under prebiotic conditions also modifications such as T6A can be obtained. So we were wondering in the end, is it actually possible to establish a primitive peptide synthesis cycle using these tRNA modifications? So having this question in mind, if it is possible to establish a consecutive RNA template peptide synthesis, we proposed a simple synthesis cycle using 2 short RNA strands that carry the modifications that we have selected from the tRNA. So in the first step, we take both of the modifications, incorporate into oligonucleotides, as you can see here as these colorful bars. In the first step, both strands need to be annealed to bring the reaction center in close proximity. And then in the second step, the carboxylic acid can be activated with some chemical activation reagents, and thereby forming such a hairpin type intermediate in which the carboxylic acid and the amino group are coupled in an amide bond. Then in the third step, we were investigating if this urea moiety, this connection to the nucleobase, can be cleaved under decarboxylation. So first thermal cleavage forming the isocyanate, and then afterwards decarboxylating it to have an amino acid transfer from one oligonucleotide to another oligonucleotide, making it possible to have several cycles here. So let's have a bit of a closer look on the coupling reaction. To investigate the scenario, it required first the chemical synthesis of the mentioned modifications. Therefore, we have decided to use standard phosphoramidite chemistry, starting from the nucleoside and incorporation of the modification by an automized RNA synthesizer. So the so-called amino acid donor strand is here a 7-mer sequence carrying the modification at the 5' end, and the so-called amino acid acceptor is having the modification at the 3' end and is a 11-mer sequence. So the first step is the activation of the carboxylic acid. Therefore, we have tested a variety of activators. First, we have tested some chemical activators such as ETC sulfonate or DMTMM, but also some prebiotically plausible activators such as methyl isonitrile or DTT in the case of using the aminonitrile derivative over here, have been used, and we could see product formation with all of these activators. Here we have a chromatogram of the reaction. So we were mixing equimolar ratios of the oligonucleotides, then adding an excess of the activator, and afterwards analyzing the reaction by HPLC. Here in the chromatogram we can see 3 major signals. 2A refers here to this acceptor strand in red. 1A refers to the amino acid donor strand in blue, and here the purple peak is the formed product 3A in this case. The second step that we were interested in is then afterwards the cleavage of the urea moiety, allowing a transfer of the amino acid. To do so, we were heating the isolated product to 90 degrees Celsius, observing already after 6 hours product formation of 15%.
5-minute warning.
All right. When we were heating for longer times, then we could also observe degradation of the RNA strand. RNA itself is not a too stable molecule. So at the same time, when we want to cleave one bond, at the same time also the phosphodiester is hydrolyzing. So as soon as we proceeded with the reaction for a longer time, also the RNA strand began to degrade further. We could overcome this stability problem by introducing modifications at the backbone of the RNA, such as 2' methoxy modifications, which enhance the chemical stability of our RNA, allowing to obtain even higher yields than 15%. Even around 80% cleavage yields were then possible. Additionally, we have tested the scope of the peptide bond formation using a variety of different amino acids, in which we can see rather similar yields in both of the cases. The major difference we can see here between the different acceptors that we were using. In this column here, we were using 2A as an acceptor, so in this case a secondary amino group. And in 2C, we were using an acceptor that is already carrying an additional amino acid, thereby having a primary amino group. And there we can observe that coupling with a primary amino group gives higher yields in the end compared to coupling with a secondary amino group, which is from a chemical perspective a bit counterintuitive, because in principle, a secondary amino group should be more nucleophilic and thereby also more reactive at the same time. But we believe by adding this additional amino acid, the distance between the reaction centers is a bit closer, which means that, which could explain the higher yields that we can observe here in this coupling reaction. Additionally, we performed competition experiments by not just using 2 oligonucleotides, but by having a mixture composed of 3 oligonucleotides, highlighting the importance of sequence complementarity. In the first example, we were using 1 acceptor strand, which was an 11-mer, having a competition experiment with 2 different donor strands. The first one having a shorter sequence, the other one having a longer sequence, and then very straightforward, of course, just the product formation with a full complementary sequence was observed in this case. A similar approach was made when we were using mismatches in one of those strands, which totally prevented any product formation having 2 mismatches in the oligonucleotide sequence. To further add a higher complexity, we were also investigating simultaneous coupling reactions, thereby having oligonucleotides carrying 2 modifications in a single strand, and also coupling again with 2 oligonucleotides, and also vice versa, having 2 donor strands and 1 acceptor strand carrying 2 modifications. Again, product formation could be observed in very satisfactory yields of 35%.
2 minutes.
Yes. So I will summarize now. Having these investigations, we were able to establish a consecutive RNA-templated peptide synthesis, yielding such nice molecules, as you can see here, composed of RNA and peptides that we call chimeric molecules, which could have an evolutionary advantage, having both moieties, amino acids, and RNA covalently attached to one another. So with these words, I would like to thank the whole Cavell group, of course. All of these people here really had a major contribution on both of these topics. And especially I want to thank Thomas, who is unfortunately not here today. And with these words and this very nice view of Munich, I would like to thank you also for your kind attention. And thanks to the funding and the whole group, of course.
All right, wonderful talk. Thank you. We can squeeze in one question. I see that Saidul has his hand up.
Hi, Felix. That was a wonderful talk. I just wanted to ask you. Well, one of the things you just mentioned towards the end of your talk was you were wondering why the N-methyl didn't work as well as the valine coupling.
Yep.
But the valine coupling is the valine substrate is actually an amino amide, so it might be that the pKa of that valine amine might be lower than the N-methyl. So maybe that's why it worked really much better than you probably anticipated. The pKa of that might be around 8 or something like that. What I wanted to ask you was, when you were doing the sort of the EDC sort of couplings to do and the other activating agents. If you didn't have the sort of the sulfur N-hydroxysuccinimide, would you have seen racemization occurring at the N terminus, or could you?
We have also tried it without the sulfur NHS, and we couldn't detect any racemization at this point. Okay. We were mainly sticking to this procedure that also was published by David Liu at the time, who of course had a different bit different background or different idea behind performing these coupling reactions, but there we were adapting his procedure, and he was using this sulfur NHS, which is a very convenient method for the coupling. Then for the prebiotically plausible coupling, then of course the methyl isonitrile or the coupling with the DTT are the better choice for sure.
Yeah. Well, the I was pleased to see you using the thiol-catalysis there, because it's quite a nice reaction. It's a simple one, so yeah. Anyway, thanks for that.
Thank you.
All right, great. Thank you, guys. We do have to move on, but please stay for the discussion. Our next speaker is Jason Greenwald. Please go ahead if you can share your screen.
Right, I'll try that. The correct screen showing?
Perfect.
Okay,
yes.
Great. Well, thank you, Irene and Nita, for this invitation. And thank you for not putting me too much later. I'm starting to feel the time zone difference here. So what I will try to do is tell you a little bit about our work. I probably will run out of time, so the punchline is a bit in the title. So for the last decade or so, our group has been working on this not always popular hypothesis that amyloids, which are a type of peptide aggregate, could be involved in the origin of molecular complexity on Earth. And we've tried to make things a bit more complex recently. So our newest result is where we work with a protocellular compartment and show that the combination of this de novo peptide formation in the presence of the, this case, fatty acid protocell creates a higher degree of localized order and complexity than you would expect independent of that, of the protocell. So yeah, we don't need to spend much time talking about this, but I like to think about, since we don't really have a definition of life, we can think of things that it usually does, and that is, or what it always does, is it's taking energy of low entropy and doing something with it. Eventually puts out high entropy energy, but in the meantime, it's creating this high molecular complexity, which we see as a high degree of local order. And there's a lot of other things that it's doing. It's self-sustaining and it's adaptable to its environment. And when we want to then think of how we want to work on a system that might be an early protocell, we have this vision of a cell. We know it's definitely not looking like that, not so complex, but we've thought about, in this talk in this series as well, but we've all thought about why do we need a cell, and it's clear that the separation from the environment and is one thing that's important. And throughout history, there's been several ideas about what a protocell could be. We've chosen to work with fatty acid vesicles, and I won't go into the history of all of these, but I want to point out that fatty acid vesicles on their own, which are just composed of relatively simple molecules, and can be like decanoic acid, they exist as many phases, so they themselves can be rather complex. We're mostly interested in the vesicle. Let me put my pointer, sorry. But there's always micelles, the vesicle bilayer, and you can have very high sensitivity to pH, salt concentration, which can change the system as well. But now I want to spend a few minutes talking about what is an amyloid and why have we been interested in that. So there's a history of amyloid, which is that they were discovered many years ago, 100 years ago, as a bad folding of a protein, and they were associated with diseases. And more recently, we realized that there's a lot of functional amyloids. So the definition is simply put, for me, is this biophysical definition, is that any polypeptide that polymerizes to form a cross beta structure. What we mean by cross beta structures, you have a beta strand here, 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 and these run perpendicular to the length of what would be a fibril that these form. So amyloids are typically then forming something of a fibril. And if you look 90 degrees rotated view of that, you see the beta strand, and it's usually packed against other fibrils in a very tight way. So this would be like a protofibril forming together, altogether sort of a larger fibril structure. And something else that we've thought about in the history of amyloids is that this was a misfolding, and you had amyloids, amyloidogenic peptides forming amyloids, and it was just a dead end, and that was usually the cause of the disease was loss of function or something like that. But now we realize that there's often a dynamic equilibrium between amyloids and their folded states, and this can be something related to their potential function in biology. So already 20 years ago, there was quite a lot of this information here. No, and this is basically what we knew amyloids could do as a function in biology. And I will kind of skip over this quickly because I will run out of time, as I said. I want to point out that it's one of the clear things they do is work as conformational templates or a chemical template. And this we've pursued further as potential for their self-replication and their adaptability, which has been shown in prion diseases that they can change their structures to adapt to the environment, is something that could be interesting in terms of evolution. And just a quick example of that, this is so-called amyloid polymorphism. This is the sum of the many structures that have been determined recently for alpha-synuclein, which is a protein associated with Parkinson's and other neurodegenerative diseases. And you see that it can this is kind of looking down the axis of the fibril. You have monomeric, dimeric, trimeric fibrils, and they can form one structure with two different types of interfaces. It's kind of all over the place. And this shows you that there's this, the energy landscape for their folding can be rather complex, and may be a way that a single peptide could adopt many different functions depending on its environment. The peptides I'm going to talk about now are generally much shorter than alpha-synuclein, so we'll talk about 5, 6, 7, 8 mers. These are also peptides that are also able to form amyloid, and they will probably have a less complicated folding landscape. But why are we interested in them in this theory? So why do I have this amyloid-centric view of the origin of molecular complexity? Well, amyloids are rather stable, and they're often more stable than the peptides themselves that they're made up of. They contain heritable information both in their sequence and, as I mentioned, in the structural polymorphism, because amyloids can template their own replication, conformational replication. And I'll talk about a few other things we've shown that they are plausible. Many groups have shown that they have many catalytic activities. And I'll talk briefly to show how they can be self-templating. And then I will get to finally the point of the talk. So briefly, amyloids form very easily when you allow peptides, especially some prebiotic amino acids like glycine, alanine, valine, aspartic acid, to polymerize. You might not say these are prebiotic conditions. We use carbonyl sulfide activated amino acids, or CDI, carbonyl diimidazole activated amino acids, and they form a precipitate, even though you have a relatively high glycine content, which is often solubilizing the peptides. But if you look at the precipitate, it is amyloid-like. It has fibrils, it's beta structured. And so we feel like that just how simple they form makes them somewhat relevant prebiotically. And already there's a selection that's occurring. So the black bars show you the initial composition of amino acids we put in, and the blue ones show the composition of the amino acids. We can't sequence the peptides that are in these fibrils, but we can, by mass spec, get the composition of them. And we see that already you have a selection against aspartic acid, against glycine, and for alanine and valine. This is not at all surprising. It's what you'd expect, because those are the more amino acids that would lead to, that are more amyloidogenic, and would be selected for in this aggregate. So one big question that was actually what sequences are made, and that's something we haven't been able to address directly, but we can think of two mechanisms, or I can think of easily two mechanisms how some selection might occur. And one is that you have random monomers polymerizing into random peptides, and then something selects them to aggregate, and that could be some similarity in sequence and amyloidogenicity in solubility. But this could also occur in sort of a solid phase templating. Once it forms, you have this acting as a template for synthesis. And we investigated that this idea and showed that it can in fact work by developing an amyloid that forms between two different peptides, this Fe repeat, phenylalanine glutamate repeat and phenylalanine arginine repeat. And we sort of imagine that the amyloid sort of stacks up like this. And since one of the peptides is missing a phenylalanine in terms of the perfect match, we have this gap here. And actually, we would expect that the fibril would look something more like this, because you have this hydrophobic patch here. So it's probably two protofibrils coming together. And this structure, we haven't yet solved. We suspect it looks like this, but this peptide complex that forms an amyloid is very selective for the addition of L-phenylalanine into this L-peptide. So it's stereoselective, it's sequence selective. It prefers phenylalanine over all other amino acids, although valine as a hydrophobic amino acid works somewhat well. And we also tested several other peptide. If we replace this arginine with a lysine-like residue, ornithine, you actually have then regioselective addition. So instead of adding onto these other amines, which would make a rather complex molecule, you still get the N-terminal addition of amino acids when you add activated amino acids to the system. So this is a sort of a cross-templating of sorts, and we've shown that you can we've created a sort of cross-templating autocatalytic pair, whereas this molecule can be templated by the red molecule to create the blue molecule, which acts as a template for this red molecule, and they can cross-template each other. We've also shown that under the right conditions, you can get a single amino peptide to catalyze its own replication. I don't have time to go into that. So now I want to finally come to the compartmentalization. We wanted to know, well, what's going on in bulk solutions, maybe rather different than what's going on when you have selection of and protection inside of a membrane. So as I said, we're working with fatty acids, and it's quite likely that any early protocell would likely depend on passive nutrient uptake. So we can work with a membrane which allows molecules to pass through it, but we want to know, how do they pass through with time? What's the flux? So we developed a diffusion-based NMR assay, which allows us to measure just the concentration of the inside relative to the outside, and that's these proton signals of the different amino acids can be measured as a function of time. And with that, we can measure sort of the permeability of valine being much more permeable than alanine or glycine, and aspartic acid being totally impermeable to oleic acid, in this case, oleic acid micelles, vesicles. We can compare this amongst different systems. So POPC, a diacyl lipid, is less permeable overall, but still permeable to those 3 amino acids. And then decanoic acid, which is a shorter chain, is somewhat permeable, but very little, to aspartic acid, but much faster permeation of glycine, alanine, and valine. So just the membrane itself is already acting as somewhat of a selective barrier for the amino acid transport, and that's something I'll come back to later. So here's the kind of outline of a few experiments I want to show. We start with encapsulating something or not. In this case, the squiggly line represents this peptide. It's a soluble peptide at neutral pH, valine, aspartic acid, repeat. And then we dialyze these vesicles that have the peptide inside and outside, and we keep just the peptide on the inside. We add activated amino acid to the outside, represented by these red dots, in this case, phenylalanine, because it has a nice signal by HPLC. And then we either dialyze or not dialyze and look at these reactions later. And I won't show you, I mean, the HPLC traces here, in the interest of time, I'm just going to point out that what I showed above here is correct. So the molecules that react with phenylalanine on the inside get trapped inside, and they don't go away. Whereas triphenylalanine, diphenylalanine, these are permeable to the membrane, they get washed away. So this peptide that I started with, I should point out, is impermeable because it has so much charge, although it's also rather amyloidogenic, we'll say. If at low pH, pH 3, when you can start to protonate the aspartic acids, this will be insoluble as an amyloid. But at neutral pH, it's the charge repulsion keeps it soluble. So we did the same experiment as above with many additions of valine this time. So we added valine continually. It was going on to the N-terminus of that peptide. And by HPLC, we can see the different products forming. Then they kind of disappear because they get too hydrophobic. And what we see is that there is, by cryo-EM microscopy, we can see these structures forming inside, which we believe are new amyloids formed from this peptide with valine added to the N-terminus. We see there's also beta structure in the aggregate that's formed inside of these vesicles. So yeah, this is just a movie to prove to ourselves that, and I can't get it to start, so that's going now. So this is, we did cryo tomography, which allowed us to look sort of inside the vesicles, and you can see all of the those lines we saw, they're actually on the inside. Many vesicles have many of these sort of lines inside, which we then can model to create this image of where the fibrils are inside the vesicle. And they appeared often to touch the edge of the membrane, which means maybe they grew to that point or they started there. Something I would like to investigate further is because there's a lot of information or a lot of knowledge about how amyloids do interact with vesicles. So another way to that the vesicle could be acting in an interesting manner is it can create a environment which protects a reaction on the inside. And so this is just to show that that would work. We set up an experiment where on the inside and the outside, we had the same peptide, starting peptide. And on the outside, we had an impermeable amine. In this case, we used aspartic acid because we knew it was impermeable. And then we added on the outside an activated amino acid, valine in this case. And valine can permeate. We didn't do any dialysis though. And normally without dialysis, you would wind up getting amyloids on the inside and outside because it would react with the peptide here and here. But in this case, the aspartic acid sort of acted as a competitor to any buildup of a reaction on the peptide here. And only on the inside did you form enough additions to this peptide, to the DV peptide, to create an amyloid. And this we also showed by cryo-EM was working. So this is just a case where the membrane is acting as a protective barrier to allow the more sort of ordered structure to appear on the inside and not on the outside. Now, finally, I want to come to what I think is the sort of most interesting point. While we were doing these analyses I showed earlier, we were also looking at what's the permeability of the activated amino acids. And what I showed here, aspartic acid in blue, is completely impermeable to oleic acid. If we added the CDI activated aspartic acid, over time, it was getting inside of the vesicles. And we had a signal for it. We also had the signal for the first intermediate, which is the N-midazoyl carbonyl asp, and that was not permeable. So we thought about this for a while, and we don't have absolute proof of this, but what makes the most sense is in the mechanism of aspartate activation in water is you have first the loss of this first imidazole when you form when the amine attacks the carbonyl, forming this compound, this N-midazoyl carbonyl aspartic acid. And this is impermeable still because it has the negative charge here and on the side chain negative charge, so it's not going to go through the membrane very easily. And then with the loss of the second of imidazole, you form this N-carboxy anhydride or Loix anhydride. This is possibly a common intermediate with some other prebiotic activations, like with I won't go into. So hopefully this is relevant. And then that can react with either water to regenerate aspartic acid, which I think we were seeing on the inside, or it can form a peptide, which we also see on the inside. So what we believe is that this molecule, which has only a single negative charge and is what's actually going through the membrane, and it is formed in such low concentrations and so reactive that we don't see it in the NMR experiments. But we do see that aspartic acid gets through when it's been treated with CDI. So that's kind of interesting. So in other words, in pink here are the impermeable molecules, whereas this intermediate is permeable. And this raised an interesting possibility. We could create a system where you essentially build up the concentration of aspartic acid inside or aspartic acid peptides, and they can't escape. So they can get in, but they can't get out. So what we did in this case is we started with empty vesicles. And we added on the outside activated hydrophobic, in this case valine, and charged what I call anchor residues, in this case aspartic acid. And when they're activated, they can get in.
Jason, 5-minute warning.
Thank you. They can form peptides. If this is done under sort of this continuous dilution, which we do with dialysis, you retain only the amino acids that formed peptides on the inside, or that if they're aspartic acid, in this case, aspartic acid will get stuck inside. And if you do this over several cycles, you eventually build up enough peptides that are hydrophobic enough that they do form fibrils. So these are amyloid fibrils that are formed de novo. So we didn't supply a template. We did bias this. In other words, we used a lot more valine because we were trying to get this to be hydrophobic enough. But these amyloids, which in principle are made up of peptides of different sequences, but maybe similar sequences, formed spontaneously. And what we can say about them is there's a high order there. There's a high local order because it's just on the inside. And this, as I say, happens only in when you have the presence of the vesicle and you do it under dilution. So this is just a compositional analysis done by mass spec of the ASP versus peptide length. And only in this experiment I just told you about, do you get these longer peptides with a decent amount of ASP. If you do it without dialysis, you wind up getting only peptides that are formed are much smaller and have less aspartic acid. So this is sequence selectivity going on in this type of reaction. Now, we can't analyze the sequences yet, and I would love to be able to do that. And if there's questions, we can talk about how one might do that. But I just want to point out that it's a system that's complex enough to where we are struggling with how we would analyze that, especially since not a lot of material is made in these reactions. I will, I think, stop there, especially since my computer seems to have frozen. Are you still on the same slide? Okay.
We can still see slides, and yeah.
Oh, that's good. It froze right before my conclusions. Let me see if I can advance it any other way. Oops. Okay, well, the conclusions are, what? I lost my mouse too. Okay, it's cool that you can hear me. So Yeah, essentially, I wanted to show that, yeah, amyloids are an integral part of biology. We know that. And it seems that biology is taking advantage of them in many ways. And we believe that they were possibly there, early on before life because of their simplicity and their stability. And now this work we've done with fatty acid vesicles raises this question of how can the membrane play a role in terms of this organization that we see occurring and the selection of sequences that we hope is occurring, but we need to pursue with more complex analyses. I hinted already that we want to analyze that. We have used some NMR and to get ideas of the N-terminal sequences that are forming, but with these thousands of potential sequences, that's not trivial. I would like to acknowledge Saidul, who did a lot of the work, Vitek Kwiatkowski, and Parik Grubic. This is work done in Roland Wieck's lab at the ETH Zurich. I have my mouse back. So anyways, I'm happy to take questions now, or maybe later. I'll try to stay up as late as I can. Saidul,
yeah, well, it's kind of late there, and we are at 2:52. But I think since it's so late, how about we have some questions, and I can try to keep my talk a little shorter. So Saidul, go ahead. Is there anybody else other than Saidul who has a question?
I'll let someone else go instead if it.
I'm just in case there isn't, you can jump right in. Anyone else?
Sudha had posted a question in the chat.
Oh yeah. Sudha, you want to speak up? Let me see chat.
Yes. Nice talk, Jason. I just wondered something about what you said in the what you showed in the cryo-EM experiments. You seem to be using POPC, so I wonder how you have the activated amino acid actually cross the membrane into the POPC. I understand fatty acids work great, but POPC, I'm not too sure. So if you can just tell us a little bit about how that experiment works.
Okay, sorry, I was a little bit distracted with my computer acting up.
She's asking whether you use POPC or fatty acids for these experiments where you get the amino acids crossing into the thing.
Okay, so we used both, and we used POPC and fatty acids, and it was basically they both work well. It was always easier to work with POPC because it's stability. Fatty acids over time in a lot of dialysis, they tend to disappear, or there's problems with them, but it works with both.
Okay, I think he's asking about the permeability of the POPC. They're supposed to be relatively impermeable, right? So how are you?
They aren't. I mean, I don't know. They're impermeable maybe to nucleotides, but to amino acids, they're fairly permeable in the sense that you have the time frame was longer. It took 81 minutes for half the valine to get in. It takes more hours for alanine. These are experiments done overnight. So, and this will, the rate at which they fill up will have to do with their size. There's a surface to volume ratio here that I'm not talking about, and these were not monodispersed. But point is, they fit pretty well to a single exponential, showing that's not too important. But no, they are permeable to many things. But they have to be fairly small, and that's why they're not necessarily the most interesting prebiotic membrane, because they do need to be so small when uncharged molecules.
Okay, thank you. Saidul, could you put your question in the chat?
I'll stick around.
Yeah, maybe Jason can take a quick look.
I'll do that now, and I'll pass now. So,
thank you. So I guess I'm the next speaker. Oh, am I? Yes, I am the next speaker. So let me launch my presentation. Having trouble with that. Strange. Oh God. It's asking me, I know what's going on. I got a new computer last week and it's not set up to share screen with Zoom yet. Wonder if somebody else should, if we can swap speakers.
You want me to switch with you?
First? Yeah, if you would go first, I can then set
no worries.
Thank you so much.
Not a problem. Yeah, I've had that before. It's a huge pain in the butt. Okay, let me switch to my slides.
Nita, if I just can add that if you can change your privacy settings, that everything will work. Thank you.
Awesome. So thank you so much for the invitation to come and talk today. I am definitely one of the people who's a bit on the outside, I think, of some of the topics that we've been talking about today. And I was really delighted when Irene asked if I would come and talk. So I'll be telling you a little bit about our work studying phage ecology. So phages are viruses of bacteria from the oceans to the human body. I want to start with my acknowledgements. This is my lab at Albert Einstein College of Medicine in the Bronx, New York, and I will be calling out individual people as I go through. I want to briefly acknowledge our funding sources and also the Google Research Innovators Program, which has provided some of the computational resources for some of the work that I'm going to talk to you about today. So our lab as a whole studies the microbial ecology of the human body. So we study microbiome interactions with the body, with chemistry, with phages, and our work is always in conversation with environmental microbiology. And that in part is why I was so excited to come and hear all of the spectacular talks here today. I'm learning a lot about how you guys are thinking about work and trying to think of ways that maybe we would be able to work together. So I'm not going to talk about this today, but we've had a lot of talks about chemical interactions. And so tomorrow, this paper from Wolfson, Hitchings, and the rest of my lab will come out in Nature Metabolism, and it's about microbially driven redox chemistry in the gut. And so I would be delighted to talk about this with anyone who's interested. But today, what I'm going to talk to you about are phages, drugs, chemistry, and health in the vaginal microbiome. So I want to start by motivating our work in the oceans, which is really where we've sort of learned a lot of our lessons about how phages, these viruses of bacteria, are interacting with bacteria and how these interactions are changing the sort of environmental conditions that these organisms are living in. And so there are sort of two main points that I want to bring up in the talk today. The first is that we are not very good at identifying phages. So there is a lot of sequence diversity out there, and it's so bad that it actually hides globally abundant groups of phages from view. And part of the problem here is what I'm calling the tyranny of alignment, that in order to be able to identify things, we generally use sequence homology-based approaches. And what that means is that we only know how to look for things that we've already seen before. And this is a problem because phages are extremely, extremely diverse in sequence. Phages also have a lot of different ecological strategies. So there's strategy diversity as well in wild phage host populations. And in some cases, this is hiding from view because of how we do experiments on these complex systems, cryptic interactions that I think we are missing in how phages and bacteria are interacting in a lot of wild populations. So I want to start by briefly telling you a story of discovering novel viruses in the oceans. And this work was done as a collaboration with the spectacular Catherine Kaufman, who was then in Martin Polz's lab while he was at MIT. Catherine has now just started her own lab at the University of Buffalo School of Dentistry, and you should keep an eye out for her because she's going to be producing some really spectacular stuff looking at phage ecology in the oral microbiome. So what Catherine did for this work was 3 months of daily sampling in the marine littoral zone, that picture that I just showed you, off the coast of Nahant, Massachusetts. So she did this every day, mostly on her own, including in the middle of a hurricane. So if you have any question as to how dedicated she is to the cause. Once she got all of those samples back to the lab, she exposed 1,300 Vibrionaceae. These are common coastal heterotrophic bacteria, isolates to concentrates of co-occurring viruses. We then sequenced about 240 of these viruses that were randomly selected from about 240 different plaque-positive hosts. So all of this was done on plates, and plaquing interactions were how we determined whether or not a phage could kill a host. And one group of viruses didn't look like anything that we've seen before. And this work was published a couple of years ago. So we had uncovered this major lineage of non-tailed double-stranded DNA viruses that were unrecognized killers of marine bacteria. The capsids of these viruses were homologs of double jelly roll or DJR capsids, which are well characterized in eukaryotes and archaea, but were almost absent at the time that we published this paper from bacteria. And so Catherine named these things the Autolycoviridae after Autolycus, a character from Greek mythology who was a thief and storied for being very difficult to catch, just like these viruses were. These viruses are quite small. They are 10 kb in length for a double-stranded DNA viruses. That is pretty small. But more of interest to us was that they have this very different ecological strategy from the other phages that had been well characterized in this Vibrionaceae system. So Joy Yang, who was a co-advised student between me and Martin and is now working for Google, put together this beautiful image where I'm showing you on this phylogenetic tree, on the ring on the outside, you can see all of these different Vibrionaceae isolates that we're looking at, these different groups of Vibrios. And each of those Vibrios has connected to it one or more phages. So these little blue dots are these tailed phages, and the gold dots are the Autolicuviridae. And it's easy to see just looking at this plot that the autos are reaching all the way across that tree in stark contrast to the tailed phages, which tend to infect one or a closely related set of Vibrios. And so these things have a very different ecological strategy that hadn't been appreciated. And when we try to do modeling of phage-host interactions in populations, it's important to know about these different kinds of strategies. We found that these bacterial non-tailed virus capsids are really widespread across the sort of bacterial tree of life. And so here we are using these kinds of sequence homology-based approaches, hidden Markov model-based sequencing approaches. And you don't have to just take my word for this. Eugene Koonin and Uten and colleagues, shortly after our paper was published a paper in which they agreed that these viruses are really a substantial component of the prokaryotic virome. And so why are these things so hard to find, right? So one of the things that Catherine noted immediately is that experimental protocols for virus preparation miss them. But to compound the problem, computational protocols for virus identification also miss them. So why is this? These things are structurally similar. I'm showing you the capsid protein here for these viruses, but very sequence diverse. So if you look at the structural alignment of two capsid proteins from these DJR viruses, these are 18% amino acid residue identity. And if you take 6 structures with significant alignments from the PDB, you can see less than 10% sequence identity in the aligned regions. And anyone who does, comparative protein structure modeling or does any kind of, homology transfer annotation will tell you, oh my God, those numbers are terrible. You're never going to be able to identify what you're looking for. You're just going to get a bunch of false positives because you have such low sequence identity. And so one of the things that we wanted to try to do was take these pieces of information that we knew about these viruses and try to ask whether or not we would be able to use our matrix of the virus-host interactions to think about what the structure, like how the sequence was related to the structure in this wild population. So this is what I mean by in the context of natural diversity. Again, these viruses and their hosts were all isolated from the same samples of water. This work was led by Catherine Kaufman. She got an assist from my postdoc, Will Chang. And so one of the first things they wanted to do was look overall at this phage-host interaction network. So what you're seeing here are our bacteria. That's our Vibrio bacteria. They are on the y-axis, so up and down. And then across on the x-axis, you can see our different phage types. So there are 4 different sort of types of phages, morphotypes of phages here. The autos are the purple guys on the left. And you can see immediately, just like I showed you in that tree, that they have sort of a different pattern of infection. So we put a little dot there in this matrix if a particular phage can infect a particular host. Next to the autos are the myoviruses, potoviruses in yellow, and then the cyphoviruses in green. And one of the things that you notice immediately upon looking at the network is that these lytic interactions are sparse. And again, we're expecting these viruses and these hosts to be seeing each other in the water, and yet these lytic interactions are sparse. Phage predator loads are low for most of the hosts. So most of the hosts, you don't have like a million phages infecting them. You have a smaller set of phages. And most phages are host strain specific. And so this was kind of confusing to us because we were thinking, we assume that there are these interactions going on between these co-occurring groups of viruses and hosts. And what we decided to do was then ask, okay, how similar is our phage-host interaction network, meaning can I infect and kill you if I am a phage for a particular host? That's what I'm showing you on the left. It's just a rearrangement of what I was showing you in the last slide. And here you can see, again, the matrix is pretty sparse. But then on the right, we ask, how much DNA do you share with other phages in this group? And so here we get something interesting. There is a lot of DNA sharing, including across the phages that we never observe being able to interact with the same host. And this is pretty weird because phages, we assume, can only exchange DNA if they are both in the same host at the same time. And so we think overall that this data set is telling us that there are some cryptic co-infections going on here in which multiple phages are getting into a host cell and in some cases exchanging DNA with each other. These diverse strategies and interactions really complicate our efforts to identify functional phage populations. So these two studies that I showed you in the oceans show that sequence diversity is hiding these large groups of phages from view, that there's a lot of ecological and strategy diversity, and we are blind to cryptic interactions such as co-infection without much more sort of in-depth studies of these systems. I'm going to move next to the vaginal microbiome, where we posit that the ecosystem there has some similar features. That is, we are going to be missing a lot of phages because we don't know how to identify them yet, and I'll get back to that at the end of the talk, and that we are going to have a lot of interactions in these communities. So why do we care about phages in the vaginal microbiome? We think that they're potentially responsive to a condition called bacterial vaginosis and its treatment. So bacterial vaginosis affects 21 approximately million women in the United States. The symptoms include discomfort, pain, increased discharge, odor, and the associated effects are even worse: preterm birth, increased vulnerability to sexually transmitted infections, and particularly increased vulnerability to HIV. The etiology of bacterial vaginosis is still unclear. There is no molecular diagnosis for this. It is diagnosed based solely on symptoms. So to give you a quick timeline of bacterial vaginosis research and treatment, in the 20s, what we now call BV was referred to as nonspecific vaginitis. And I did a sabbatical at the Radcliffe Institute at Harvard, and I was able to go through the Schlesinger Library, which is one of their libraries that has collections from the Boston Women's Health Collective. And I went through some of these documents to try to understand what were people doing to treat bacterial vaginosis before we knew much about what was going on in the vaginal microbiome. And so one of the things that I came across there was this. There was a lot of stuff like this where you have these pamphlets that are meant to help women understand and work on their own health. And here, bacterial vaginosis or nonspecific vaginitis is basically referred to as a domestic tragedy that destroys romance. So there's a lot of this kind of, not actually talking about the thing, sort of talking in euphemisms. The solution here is douching with Lysol. So this was absolutely horrifying to me when I found this, but this is a sort of common feature of these pamphlets. This one is from the 1940s, where there are all of these home remedies and ideas for how to treat these conditions. So in 1955, Gardnerella was isolated, and this was assumed to be responsible for bacterial vaginosis. But by the 60s, it was shown that Gardnerella was absent from many bacterial vaginosis cases. In the 70s, bacterial vaginosis started to be treated with the antibiotic metronidazole. Now in 2010, we finally started getting some community sequencing of the vaginal microbiome. And what did that tell us? It told us that alterations to the vaginal microbiome are a hallmark of bacterial vaginosis. So in healthy or asymptomatic women, the vaginal microbiome tends to be dominated, but not always, by a single species of lactobacillus. It has an acidic pH, there is low microbial diversity, and gram-negative anaerobes are present in low abundance. In contrast, everything in bacterial vaginosis is flipped. So lactobacillus abundance is usually reduced, the pH is higher, there is a higher microbial diversity, and you have a lot more of these gram-negative anaerobes. Now, I'd like to tell you that we're doing so much better than the 1940s in terms of how we treat this, but in fact, we're not. So we're still treating with metronidazole, and it has a very high recurrence rate post-treatment, in fact, 50% after 12 months. clinical response to metronidazole also differs in different populations of women. So response rates of 95% in the Bronx versus 55% in Thika, Kenya are telling you that there are some differences in vaginal microbiomes that are influencing response to metronidazole treatment. And so what we want to do is ask whether or not phages are playing a role in recurrence of bacterial vaginosis after treatment and in differences in clinical response to metronidazole. And to do this, we're working with the spectacular Betsy Harold and Marla Keller here at Einstein to study adolescent and young women with clinical bacterial vaginosis who are treated with metronidazole and asymptomatic matched controls. We're isolating vaginal bacteria, identifying phages, and characterizing bacteria-phage environment interactions. I'll just tell you a little bit about some early results here. One that surprised us a lot is that there are very similar phages across different women in this study. These are all cyphoviruses from lactobacillus isolates from the same Bronx individual in the black box. That's perhaps not so surprising. We isolated all this bacteria from the same person, and we see that some of them have the same phages. What we didn't expect was to find an effectively identical phage in a different patient. And then when we took that phage out into broader studies outside of our own, we find extremely high identity phages in Dallas and St. Louis as well. So this is interesting because these phages, the similarity of these phages is decoupled from the similarity of the bacteria, at least in our study. And so we're curious to know what is prompting what looks like a very unexpected conservation of phages across different women's lactobacillus isolates. To get back to this question of the tyranny of alignment and how we are trying to identify novel phages in the many environments that we've been working here, from the oceans again to the human body, my student Zach Flemholtz has started using protein language modeling approaches to viral classification. What he's doing is basically embedding genomic and metagenomic sequences using pre-trained language models that are based on protein sequence databases, and then using a transfer learning-based multi-label classification of viral protein families. And again, the goal here is to identify viral sequences, not using sequence alignment. So we're trying to go outside of this alignment paradigm. And so when Zach came to the lab and said he was interested in doing this work, I was like, knock yourself out. This is never going to work. And I want to tell you some of what he's done that is convincing me that I should probably eat my words.
A 5-minute warning.
Awesome. I am just about done. So this is all of the proteins from that Nahant collection that I told you about. We're going back to the oceans here. So this is all the proteins from those 241 phages embedded with ProtBERT. So again, we're not building these language models on our own. We don't have the resources to do that. We are using pre-built language models. So this is all the proteins from the Nahant collection. And then we take this spectacular database of phage protein families called frogs. And we say, okay, give us the top 10 largest capsid protein HMMs and tell us where they fall in this embedding space. And so you can see these 10 different capsid proteins and where they fall in our space. Now I'm lighting you up just the capsid proteins from our data set, which were not present in the frogs database. These are shown in red, and most have good overlap with these capsid families that we see. Not all of them though, right? You'll notice in that right hand upper part, these little red ones that look a little bit different than the others. And so now we go into Uten and Kunin, who did another paper in 2021 where they identified more double jelly rolled capsid sequences, and we asked, where do those fall in this embedding space? And when we compare those to where our auto sequences are from the Vibrio data set, we can see that by sequence alone, our capsids are falling into similar embedding spaces. And so this is really early days with this work, but I am enthusiastic, and I think it's worth continuing to work with this and see if we can do a better job of identifying viral sequences that we don't already know how to look for. And so in summary, when we're thinking about phage ecology in the human body, particularly in the vaginal microbiome, we're interested in what conditions activate phages. So we talked about antibiotics. In some systems, the antibiotic clindamycin has been shown to induce phages. bacterially produced compounds. So sulfide, for example, the subject of our other publication that's coming out tomorrow, we are curious to know if sulfide itself can also act as a phage inducer here. We know that there are pH changes. What are other environmental changes that might be activating these phages? And we, again, same as in the ocean, this time we want to think about the structure of phage-host interactions in the context of natural diversity in the human body. And with that, thank you so much. And I am happy to take questions.
Terrific talk. We do have about 2 minutes for questions. People want to put them in the chat or raise their hands. If there aren't any questions, I'm happy to start. Okay. Oh, there's a question in the chat. Okay, great. From Zach. Thanks. From the first half of your talk, patches of shared DNA seem to show up in sparse areas of phage infection. The 2 cross-plot heat maps that you showed, is this significant?
Is it significant, meaning did we expect to see it, or do you mean based on, it is significantly different here. I'll just show it to you. It is significantly different from
Zach, you can chime in if
the
you
interaction
want to clarify your
matrix
question.
here.
Yeah,
So
it was
yeah, these are
just a
significantly
pattern I
different in
noticed.
matrix
I
properties.
wondered if there's a mechanism underlying that, or if it's just kind of a feature that you expect to see in a relationship like this.
This was not a feature that we expected to see, because we expected that the one on the left is reflecting reality, that this is who can infect who, but the one on the right suggests that there are more infections going on than we can see.
Got it. Okay, I think I understand that now. There might be a whole other layer of interaction that's just kind of being missed by this. Oh, that's even more fascinating. And then for the second question, for the protein language model with the BV phages, you mentioned that you're breaking it into this classification. How are you translating the sequences into what are the basic units of the language that you're analyzing for?
Yeah, so the Prot-BERT model is based on a combination of Swiss Prot and EMBL sequences. And so what we're doing is just straight up feeding the sequences of our phages into that model. And so then we're using a neural network effectively to do the classification. And the nice thing about the neural network is, well, with other machine learning techniques, you can do this as well. But for us, it's nice to know how sure is the model about where that sequence falls. And I think there's a lot to be done to sort of tease apart how do we, assess our certainty and how useful are these models.
I'll just bring in, I think we may not have enough time to get in the next one. I'm fascinated because we're also struggling with how to apply ML approaches to making sense of other chemical network interactions. And we're facing exactly the same problems, but completely shoved into a chemical reaction space as opposed to an ecological space. So
yeah,
thank you so much for your talk.
We should chat. Seriously, this has been, and Irene, thank you again for the invitation. This has been really cool for me as well.
Yeah, I hope we can all stay for the discussion. But we should move on. Great, thank you. Nita, are things working now?
Yes, thank you.
Okay,
So,
great.
all right. Are people seeing my screen?
We're seeing your whole screen. Oh, that looks great. Perfect.
Good. Well, I guess I'll launch right in here. Yeah, so what I'd like to talk to you today is about our work using minerals as catalysts, prebiotic catalysts for coevolution of biological or proto-biological molecules, really. And also selecting those specific molecules, like why did biology choose alpha over beta amino acids, for example. And also to show you how minerals might have catalyzed protometabolism. So here's a picture of a Hawaiian hot spring. It'd be nice to be out there right now, but the idea is a surficial environment, most likely a volcano sticking out of the ocean somewhere, whether or not there were early continents. So this idea of synergism and mutualism in the coevolution of biomolecules was proposed. We proposed that some 8 years ago. And the idea is that this coevolution occurs under environmental conditions such as wetting, drying cycles, the presence of catalytic or non-catalytic metals, catalytic or non-catalytic minerals, the temperature, some energy input where that wetting drying cycle provides the energy input. So I'm going to show you a few examples here where we talk about mineral-catalyzed RNA polymerization, which is co-catalyzed by amino acids. So there's coevolution of those types of molecules. And then also the formation of a novel kind of a proto-membrane forming molecule with amino acids and fatty alcohols involved. And again, finally, mineral catalysis for metabolism. Now, before we get into anything else, we need to first know what kinds of ions and minerals were actually around for prebiotic chemistry. And this is where I am very interested as a geochemist in trying to say there's some possible constraints on what might have been plausible on early Earth, and that's based on rock weathering, basically. What gases are in the atmosphere when they dissolve into rainwater, they form some kind of weak carbonic acid because of the carbon dioxide in the atmosphere. They fall on the surface of the Earth, dissolve the exposed rocks. That causes new minerals to form, releases ions from the previous existing rock minerals. So you get a partial equilibrium of the atmosphere, the water, and the rocks. So that sets up the stage for what concentrations of ions, what complexes, and what minerals were around. And we also do wet-dry cycles. That's going to then also, it's basically looking at like an evaporite basin, such as you might have in Salt Lake. So we published this earlier this year in Astrobiology. Where we basically looked at the weathering reactions of a komatiite-like rock, which would represent Ocean Island in the Hadean, as well as tonalite that would represent any tiny amounts of protocontinent that might have been around. We're agnostic as to whether these were around or not, but if they were, then what kinds of compositions would they have produced by their weathering? And you can see here some rocks representing the komatiite-like crust that might have been there for the oceans, and the tonalite-like rock. The difference in color reflects the difference in mineralogy, which ultimately reflects the difference in chemical composition. So these are enriched in magnesium, calcium, and a little bit of iron. These are enriched in calcium, sodium, potassium, and these are all these other elements. And there's phosphorus present as hydroxyapatite coming up from the cooling of the magma, which created these rocks. That's the source of the phosphate. Now we equilibrate the atmosphere, which is whatever concentration of carbon dioxide. People have estimated a huge number of partial pressures of carbon dioxide, PCO2 in the atmosphere for the Hadean Earth. Again, we're agnostic and did this calculation for a whole range of them. And when you do the weathering reactions, what do you get in terms of what's dissolved by dissolving those rocks? How much do you get in solution for total dissolved phosphate, magnesium, iron, and calcium? And we need some way to constrain what kind of partial pressure of carbon dioxide we're going to use and subsequent chemistry we're going to use for these solutions. So we said, let's use the 1 millimolar biological level for the iron, calcium, and for the magnesium, calcium, and phosphate as our marker. And the particular carbon dioxide partial pressure that we get from that, we'll use that for our further simulations. And for the iron, that same value is at around 10 to the minus 5 for the biological level. Now, when you so you can see that basically at about 1 to 5 atmospheres of carbon dioxide, you get about a millimolar of magnesium. When you do one cycle of evaporation of this, the concentration really drops down significantly. And the same sort of thing is happening for iron and calcium. They're way below the millimolar level. And that's because of the precipitation of clay minerals and carbonate minerals that are pulling out these cations. On the other hand, the phosphate concentration actually goes up when you do the evaporation because hydroxyapatite is not precipitating. The calcium having been removed already as a carbonate, then there's no calcium left for the phosphate to precipitate. So the important point here is that you have very low divalent cation concentrations. And so this is good because then the fatty acid protocell membranes can self-assemble. Those fatty acids are very sensitive to divalent cation concentrations. But since we show that these are so low here, then the membranes can be stable if they're fatty acid based. However, now that you've got such low magnesium and iron, doing RNA polymerization is going to be very difficult by either the RNA template method that Jostak uses or by the montmorillonite method that Ferris used. They both required 75 millimolar magnesium. So we have a problem here in terms of how to do RNA polymerization at low magnesium concentrations. The phosphate concentration is also fairly low, but it turns out that because you build it, you increase the concentration during precipitation. Every time you do a wet-dry cycle, you can actually build up the phosphate concentration. As you can see over here, as you do numerous wet-dry cycles, after a couple of hundred cycles, you can build up the phosphate concentration to fairly high values, even up to about 100 millimolar to 1 molar, which might be important if you want to do the kind of nucleotide synthesis following Pauner et al. So the take-home message is the atmosphere and the rocks around determine the relative availability of the ions and the minerals. And so what are the implications for prebiotic RNA polymerization and protocell membrane self-assembly? All right, so the question we asked is, how do you do RNA polymerization at low magnesium concentrations? And we wanted to show coevolution of the biomolecules. So here's the classic Ferris experiment where you have an activated RNA monomer. It combines with a dimer. If you just have aqueous solution, 75 millimolar magnesium, pH 7, you just get a dimer. But if you put in a montmorillonite clay, you get an 8mer. So this has been known for 30 years. How do you improve this polymerization efficiency, and how do you get this to work at a lower concentration? That's the question we wanted to ask. So what we did was, let's look at how amino acids, which would have been around in the environment, may have affected this whole reaction. And so Trishul Namani, my postdoc, looked at the addition of, here's your activated RNA monomer, here's the clay mineral, here's the classic Ferris. If you don't do no amino acids, you get a RNA oligomer of about 8mer. Now, if you add amino acids, if they contain a non-polar side chain or an acidic side chain, we saw this beautiful result that there was a promotion of the RNA to a longer chain length. Whereas if you add the basic and the polar amino acids like serine and cysteine, then there's an inhibition compared to the no amino acid system. However, this is still better. Even these so-called inhibiting amino acids are still doing better, the former, than if you had no mineral around. Remember, when you have no mineral around, just in water, all you get is the dimer. So even when you have a so-called inhibitor, inhibiting amino acid, you still get plus the mineral, you still get a tetramer. So we have a reaction mechanism to explain why these guys are promoting, why these guys are inhibiting. If you want to ask me later, I can get into the details of that. But for now, I'm going to just skip that for the interest of time and tell you that we analyze this using both MALDI-TOF as well as HPLC to determine what our polymerization, degree of polymerization is. Now, what's even further interesting is that these guys who are doing the promotion, as Jason pointed out, are the prebiotically most plausible ones. The GAD and V are the most plausible prebiotically, and they're the ones which are doing the most promotion. So this was really exciting to us. And further, we said, however, this is still using 75 millimolar magnesium. We want to drop that. So we tried the next thing is, what's the effect of these amino acid co-catalyzed montmorillonite catalyzed RNA polymerization? So when you have amino acid like glutamic acid, which is a promoter, and you have the montmorillonite clay, then how much magnesium do you need? So here's your clay, here's your activated monomer, there's no amino acid. With 0 magnesium, you just get this dimer again. Now, if you take the system and just add glutamic acid, then you get already a tetramer or a pentamer. Now you add 5 millimolar or 10 millimolar or 15 millimolar magnesium along with the glutamic acid. You get longer and longer chains. And then finally, here's that classic 75 millimolar, which gives you the longest chain when you have this glutamic acid around. So the key take-home message is, when you have glutamic acid or any of those, frankly, any of those promoting amino acids, the acidic and the nonpolar, and the clay mineral, so you have co-catalysis going on, then even in the complete absence of magnesium or at very low concentrations, relatively low concentrations, which we have seen from the geochemical calculation that I first showed you, these are actually plausible in the environment, then you're getting a decent amount of polymerization or at least oligomerization compared to simply the dimer, which you would get without any magnesium or without any mineral. This is fantastic because now, depending, you might have a geochemical environment where there's no magnesium or very low magnesium or very high magnesium, let's say, some out of equilibrium environment where high magnesium is built up. And you can get oligomerization in all of those environments. And in these conditions, the protocell of fatty acid membrane is going to be stable under these conditions. So the take-home message is amino acids with certain side chain functional groups and clay minerals co-catalyze activated RNA polymerization between low to high magnesium concentrations. All right, moving on. Now we come to this idea of selection. We talked about catalysis and coevolution, but there's a whole broth of molecules in the prebiotic environment. There were all kinds of left and right-handed enantiomers. There were configurational isomers in the case of the amino acids. So for example, we have alpha amino acids where the amine is attached to the alpha carbon, beta where the amine is attached to the beta carbon, gamma where it's attached to the gamma carbon, delta for longer chain amino acids. All of them would have been around in the prebiotic environment. Why did biology primarily select alpha amino acids, although not entirely exclusively, almost exclusively? How did that selection occur? So staying within our framework of the systems chemistry of the clay-catalyzed RNA polymerization and amino acid acting as co-catalyst, we said, let's see whether these other amino acids, the configurational isomers like beta and gamma, have the same effect as the alpha on enhancing or inhibiting morillonide promoted RNA polymerization. And so what we found is when we add beta amino acids, non-polar or acidic or or the basic and polar, there's no effect. It's as good as not having any amino acids around. So this co-catalysis of amino acid is effective only for the alpha amino acids. And that's very cool because what it means that the alpha amino acids promote the longer chain of the RNAs that presumably can then fold and have some function and then maybe reselect back in for the alpha amino acids when it comes to making a peptide. Here's the mechanism. I'll get into this later. Save that for later if people are interested. Now, we've been talking all along about synthesis, of these different kinds of molecules, coevolution. So now we want to look at the problem with fatty acid membranes is that they're fairly unstable to a wide range of pHs and cations in the environment. So we hypothesized that if you make amino acids which are present in the environment and take fatty alcohols, which are also abundant in the prebiotic environment, and do gentle wetting drying cycles, at about 50 degrees Celsius, you can form these amino acid fatty alcohol ester amphiphiles. So here's the tail that is coming from the fatty alcohol. Here's the ammonium that's coming from the amino acid. Here's the carboxy that's coming from the amino acid. So, and we've identified them with mass spectrometry. We've done this with glycine as well as with a different amino acid called isoleucine, and using octanol as our specific fatty acid. We find that they make beautiful vesicles at when we combine them with octanoic acid in a 1-to-1 ratio. these vesicles, these glycine octanoic esters, make beautiful vesicles at pH 2, at pH 4, at pH 6, at pH 10. So across the whole pH range almost, you can get these vesicles of either pure GOE or mixtures of GOE with octanoic acid in different ratios. Amazingly, these are even stable at very low pH and up to 100 millimolar magnesium chloride. Would there have been some kind of environment with so much magnesium around in it? You know, even if all my calculations from the first part of the talk are wrong, and there was some environment with like a huge amount of magnesium around, even then these vesicles are stable. And this is very easily understood because it has an ammonium head group, which is positively charged over a wide pH range, and a carbonyl, which has a negative charge density associated with it, which gives it the wide pH range stability. And of course, magnesium is not going to bind to this head group, as opposed to a fatty acid head group, which it would have bonded to. So here again, we are showing that amino acids and single chain amphiphiles could have co-evolved from very robust protocell membranes.
A 5-minute warning.
Thank you. And then some old work using fatty acids and fatty alcohols. We were interested, actually, Jostek and Hanchik had shown, like 2007 or 8 or something, that most of them, any solid surface they pretty much looked at promoted the rate of fatty acid vesicle formation, but there was no mechanism. They found no trend. So we did a similar sort of experiment where we looked at the log rate of the fatty acid self-assembly into vesicles, and we have the cryo-TEM here, in the presence of a whole bunch of different minerals, oxides and carbonates and sulfides and clay minerals, monaurolinite, even whole rocks, komatiite and tonalite, the 2 rocks I showed you right at the beginning, crushed up. And we did them for particles that are in the micron size range and particles which are in the nanometer size range. And note that this rate that we then extracted for self-assembly is normalized to the surface area per meter square. And we find this beautiful correlation with the isoelectric point or surface charge of the mineral. Experimental pH is 7, so minerals with isoelectric point over it, just like a protein, are positively charged, and these ones below it are negatively charged. Our molecule has a negative charge on it, so the vesicle is negatively charged. And so it makes sense that the more positively charged will absorb more of these negatively charged vesicles, and therefore there is a better catalysis, autocatalysis of the adsorbed lipid on the mineral surface that then makes the vesicle formation even faster. And again, we have a mechanism, can get into that later. So we were the first to identify this structure-activity relationship that depends on the surface charge of the mineral as well as on the size of the mineral. And interestingly, you can even predict the isoelectric point of a mineral. You tell me any mineral based on my PhD work, I can predict for you what its isoelectric point should be, because I know what its crystal structure is and I know what its chemical composition is. And so there's a theory for I developed for my PhD to predict the isoelectric point of a mineral simply based on its crystal structure and composition. So you tell me in any mineral, you go to any surface of Mars or some other solid planet, what's the mineralogy? I can calculate for you if a vesicle ever self-assembled there, what would have been the rate? All right, to finally wrap up, I wanted to show to examine whether minerals can photocatalyze a kind of a prebiotic, at least a part of a prebiotic metabolism. So we built a vesicle system consisting of either oleic acid or phospholipid or mixtures of them. Within it, we encapsulated NAD+ and this molecule rhodium trisbipyridinium. In the membrane, we embedded polyaromatic hydrocarbons. Outside it, we added different catalytic minerals like pyrite, cadmium sulfide, titania, zinc oxide, and we added any kind of simple organic molecule like an amino acid or any other simple organic that might have been around as an electron donor outside. And then we shine UV light on it. What we want,
2 minutes.
2 minutes, is a hole and an electron separation on this photocatalytic mineral. The hole gets picked up by this reduced organic compound and gets oxidized with the generation of protons outside the membrane. the electron that is generated in the catalytic mineral is picked up by this polyaromatic hydrocarbon, which then transfers this into the vesicle, where the rhodium tris bipyridinium in its oxidized state is then reduced. It's a 2-electron simultaneous transfer to a +1 state. Then this guy is able to give up its electrons, simultaneously 2 electrons to NAD+, which then gets converted to NADH. This is the scheme that we built, hoping that all of this will happen. To determine whether this is actually happening, we follow the production of NADH inside the vesicle by using fluorescence spectroscopy. And so when you irradiated pyrite, greenockite, etc., different minerals, at 0 hours, 1, 2, and 3 hours, you can see the buildup of the NADH with the emission at 450 nanometers. Also, we are claiming that there's proton generation on the outside, and so it should be possible to figure this out depending on if there is proton being generated outside, and if you have a very intact vesicle like a phospholipid vesicle, then none of it should leak in and acidify the interior. And in fact, we find that when you plot the pH inside the vesicle versus time for a phospholipid vesicle, it didn't really change over time. However, when you use an oleic acid vesicle, which is more leaky, it does leak in, the proton does leak in, and so the pH drops a little bit over time. So we did indeed generate a system like this with a number of different photocatalytic minerals. This one is used as a positive control. Obviously, a quantum dot wouldn't have necessarily been around.
Nita, you're out of time, but okay, last slide, right?
That is my last slide.
Okay.
All right, thank you. So, yeah, questions?
I see Moran had her hand up. Why don't you go ahead?
Stop sharing. Where do I stop
sharing? Moran, you can go ahead. We have time for only one question. I'm sorry. I hope everybody else can wait for the discussion.
Yeah. Moran, go ahead.
I am not hearing. I'm not hearing anything. Okay, let me just defer that to the discussion then. Hopefully we can fix that audio problem. Okay, because of the time, let's go ahead and move on to our next speaker. Thank you, Nita, which is Uli Gerland. Uli, are you here? Okay.
I'm here. Can you hear me? Yeah.
Yes, great.
Okay, great. Okay, let me just get my talk sharing. Worked out. So. Does that work for you?
Looks great.
Yeah. Okay. Well, so then, yeah, thanks a lot, Irene, and all the organizers for having me here. So, I'm a physicist. That's my background, and we look at systems that are sort of RNA molecules and that are interacting with each other, and so something like you see here on the left hand side, we call them RNA pools. So I guess we all know that there are sort of interesting systems out of which something could emerge, and there are lots of intriguing questions here that we can of course not address by ourselves, but we hope to somehow contribute to this. Yeah? So this is a list of these questions. For example, if you have interactions between RNA strands that are in these pools, what kind of things can emerge from that? I mean, could there be, in particular, some pathways that ultimately lead to the emergence of ribozymes in some primitive prebiotic evolutionary process? If such a function is perhaps found, through some random search of sequence space, how could that function be maintained? And yeah, so how could molecular evolution emerge spontaneously? Maybe it's not a full evolutionary process yet, but it's something sort of on the way towards that. Yeah? So that's fascinates me as a physicist, and we're wondering sort of what are the elementary processes. So this work is that I'm going to report here. So our own work is purely theoretical. Yeah? So this is a computational work mostly. And we describe systems of RNA strands as sort of a system where you really have individual sequences of letters that somehow interact with each other. Yeah? And so this is not a full-scale, let's say, molecular dynamics simulation or something like that. It's really on a more coarse-grained level. And let me try to explain what that means. That means for us, we have individual sort of elementary processes. So for example, if you just have a hybridization between two different RNA strands or a dehybridization, that's sort of a single step reaction within our framework. You could have this red strand binding to the gray one with a certain rate constant. And of course, how often this happens, so the flux or the rate depends on the concentrations. You have some finite volume with a starting condition of strands, and then they start doing this. No? Of course, that's not the only thing that can happen. We can have actually the finite volume may even be a non-equilibrium system in the sense that it's open. Yeah? So there might be influx of molecules, outflux of molecules. I should say that we assume here that processes like the ones that Nita talked about just before me, like the spontaneous polymerization that leads to RNA strands, we assume that's a given. Yeah? So that there are sort of ways to chemically energize and overcome sort of the barrier towards bond formation. Another elementary reaction for this is the spontaneous cleavage. Yeah, so basically the cutting of an RNA strand, and the inverse process, essentially, even though chemically it might not be the exact reverse reaction, is the ligation process, where two strands sort of form a longer strand, and this reaction, of course, is enormously sped up in rate. So the rate constant is increased a lot if there's a template. So you could have a template-directed ligation process, such as shown here. Of course, RNA and other polynucleotides, they also can fold and form structures, and that's super important to obtain catalytic functions. So this should be also incorporated in such a framework that can describe such systems. And you could even have processes that where sort of particular binding sites on strands are blocked by one molecule, but a second molecule comes in that somehow displaces this, which would then be a tri-molecular reaction process, such as the strand displacement reaction shown here. Okay, so this is, for me, let's say the framework that we can work with. Yeah, so we have a system that's specified by a given volume, a given starting condition, which is basically a list of, let's say, sequences. Yeah, so I've drawn them here just as little strings, cartoons here, but of course these are all sequences with letters, and then they evolve according to rules on the computer. And of course we would like to do something with it that helps our understanding of the system and sort of says something about experiments. So the question, I guess, that you might be wondering about is how can theoretical work of this type, how can it actually contribute to the broader field? Yeah. So I think there's two sort of pillars or two ways in which we can potentially contribute. Yeah? One is sort of if we have a concrete experiment. So I will give examples later on, briefly at least. And we're trying to adjust all the parameters of the models such that we fit that experiment in a certain regime, and then we can try to extrapolate from that regime where the model is tested and try to predict sort of outside of that regime. Yeah? So why is that helpful? Well, the nice thing on the computer, of course, is always that you can measure anything during the computer experiment that you would like to know. And there are certainly a lot of interactions in these systems that are just transient and happen for a short time and then produce something else and so on. So which you cannot easily observe experimentally if you have such a pool of RNA molecules in a non-equilibrium setting, such as, for example, microfluidic systems. So Dieter Braun in Munich is one of our collaborators. He does this experimentally, and so we can try to sort of make predictions on detailed processes that happen within these soups, these pools, which can then be followed up on experimentally. Now, the second type of contribution that we hope to make is that we can just on a more general level, somewhat detached maybe from experiment, initially at least, we can say, okay, if we just put a certain set of ingredients in a soup, yeah, and I'll explain what I mean by that later. Even if we don't know exactly like all the rate constant and all these parameters that of course are important to describe such a system faithfully, can we say something about what types of behavior can emerge? And that I would say is a conceptual model, yeah, that is just qualitative. And hopefully the real system behaves in a way such that the behavior of the real system is sort of somewhat robust, so that even if you have a model that's not entirely correct, it doesn't have all the parameters that you need in order to predict a particular experiment completely faithfully, that it still displays the same types of behavior. No? Okay, so this is all done in collaboration. I'm going to point out so that Irene Chen and I, we collaborated on this in the past, so in particular on the left-hand side here of the slide. Yeah? And but before I get to examples, I just briefly want to also say what are the challenges in this. Yeah? Why is this hard for us technically? And a large fraction of that is just the combinatorial explosion of possible different interactions between RNA strands that you can get, and that all the reactions that you can have, cleavage and ligation and folding and hybridization and template directed effects and so on, which are all sequence dependent, and the processes that can happen, they change after each reaction, because you create a new sequence or you destroy a given sequence, and then the list of possible things that can happen in the soup, in the pool, changes. Yeah? So even when we don't describe this like on a molecular level with full blast simulation of like the conformations of an RNA polymer, but if we just list the reactions in a more abstract space as we do, then this becomes very, very challenging, and you can guess that this will become more challenging as the systems become bigger. Yeah? So our limitation is that we cannot easily describe very large systems. They have to be small, meaning not so many molecules at the same time. Yeah? Well, that's one challenge, the combinatorial explosion. Another challenge is that we have a lot of different rate constants, and they of course depend not only on the sequences, but also on the particular condition of the experiment, salt concentration, temperature, and so on, you name it. And that is not fully known. Yeah, actually, we're far from that. Yeah? And then in addition, even if all those things had been solved, let's say, you have a huge separation of timescales in the system. So that means you have some rates that are very high, another set of rates that are very low. For example, hybridization and dehybridization are usually relatively fast compared to ligation reactions. So that means you need to describe the kinetics of these systems on the level of the fastest reactions, because those are the ones that happen most of the time. But then you also want to get to timescales where the slow things and interesting things sort of happen. Okay, so some of so what are helpful things that make some of these challenges a bit easier? So one constraint that we have, some set of knowledge, is that this complex dependence of the rate constants on the sequence that is at least partially predictable from you may know this sort of free energy rules for RNA folding and also for DNA. These parameters basically for base pairing and structure formation, they have been inferred from lots of different sort of melting experiments. So if there are sort of certain stackings of different base pairs on top of each other, there is a measured number that is, of course, dependent on temperature and dependent on salt concentration, but there are rules to, let's say, estimate these. So we make use of that. So this is sort of built into our framework. And okay, so part of our work that was done in close collaboration with Experiment, in particular with Irene also, and with Clemens Richard in Stuttgart partially, uses those free energy parameters in order to try to predict the efficiency, the speed, and the error rate of non-enzymatic copying processes. What does that mean? That means you have a template strand that encodes some information, and of course, for a molecular evolutionary process to emerge in such a pool, you need a copying and replication mechanism. And what's important in order to see whether this copying can preserve a function that has maybe been found by chance, by pure sort of random assembly of oligomers, is how faithful is that replication process. So this depends basically on the amount of errors that you make in such template directed copying processes. So this is one topic that we've worked on, and there one can sort of start from the parameters that are known for hybridization, like these free energy rules that we see here on the right hand side, and then try to adjust remaining free parameters such that it actually describes the experiments. So okay, but I want to maybe now focus and because that's it. The challenge I want to address in this talk here and talking to you is to motivate you also from the physics, for the things that for physicists are maybe more natural than in an interdisciplinary community, namely this right-hand pillar. So this right-hand pillar of our work is just exploring emergent behavior. So I guess you can all imagine how sort of fitting experiments and so on that works. But this may be more mysterious. How can we? So what does it actually mean to explore emergent behavior? So we started with this a bit more than 10 years ago, and we had a original version of our, let's say, software toolbox that we call the RNA reactor. And that had some significant limitations, which we have now overcome to some degree. I will tell you about that in just a minute, but I want to briefly mention sort of this original work because that illustrates also the idea of emergent behavior. So this original work, what it did is it had built in basically random ligation processes. It had hybridization built in. It had influx and outflux, and it assumed that all these things would be happening in a small non-equilibrium fluidic system like the ones that Dieter Braun in Munich is experimentally probing, where there is a temperature gradient that is sort of keeping the system out of thermal equilibrium, and this temperature gradient produces a convective flow and at the same time thermophoresis, and the interplay of these two mechanisms leads to the enrichment of longer strand. So this is sort of an interesting physical mechanism that just biases the system towards longer strands. So we had that built into this system, inspired by experiment. The caveat is here, there's no template-directed ligation process built in. So this is pure, just cleavage, ligation, hybridization, and also folding. So self-folding of RNA molecules. Now, nevertheless, so if you don't have template-directed ligation, you can imagine that you don't easily get a copying process. So you may say it's completely impossible to get replication in such a mechanism, in such a system.
Rick, there's a 5-minute warning.
Oh, okay, I'm going too slow. Yeah, but thank you. So the let's say the message, and I will skip the details here. The message of that particular work was that if you have a protection from cutting, so the key thing is here, so that the cleavage of an RNA strand is much more likely if it's single-stranded than if it's double-stranded. So if the cleavage of an RNA strand is base pairing dependent, then you can actually get something that resembles a bit a replication mechanism without template-directed ligation. So basically, hybridization then protects a motif, a sequence motif. If there's the inverse motif, also the reverse complement, you can protect it, and then that means the reverse complement and the original motif itself, they protect each other, and they basically are just formed by random chances, but retaining these motifs that is then happening more likely. So that means that the information stored on these motifs is maintained longer than the lifetime of the actual molecule. And that's something that I'm very interested in. I'm not sure how contrived this question is from your point of view here. So this is from a physics point of view. It's sort of the separation between matter and information. So because you have molecules in which information lives, and usually, like before information becomes a separate entity, the information dies when the molecule dies. But can the information survive longer than the actual molecules that carry them? Of course, if we have replication processes, that's easy. That's a given. Of course, you conserve the information, but without true replication, is there sort of a pre-stage? And that is sort of the main message of that particular work here, which shows that even though you don't have true replication, you have an effective replication process that is sort of weaker than the destruction of molecules. You get sort of maintenance of information over longer timescales. So it's at the beginning of the emergence of separation between matter and information to some extent. Okay, but just in the few minutes that are left, let me get the point across that. So in terms of just as a tool, if you want to use this as a tool, our RNA reactor toolkit here is sort of very limited because in this piece of work, the hybridization and the folding are assumed to be equilibrated. And we only have interactions between two strands, no sort of higher order processes between multiple strands. And we recently overcame that challenge in a sort of second version, let's say, of this RNA reactor. And I don't have time to go into the details, but here all the reactions are described fully kinetically. Yeah? So there's no equilibration assumption about separation of timescales. And also we can have complexes that consist out of multiple strands. Yeah? So for example, I guess I need to come to an end. So let me just briefly wrap this up. Yeah? So we can look within this sort of new, more capable and versatile framework at the emergence of behavior. And let me skip that. Yeah, polymerization process. So what we find that in such systems, a sort of peculiar phenomenon emerges, and this is something that is confirmed experimentally. So maybe that's the message I should leave you with. So what is shown here in this plot is the strand length distribution in such an RNA reactor. And so the x-axis shows the length of these RNA strands, and on the y-axis you get the concentration of that particular strand length. And you see multiple curves here, and these curves basically differ by an outflux rate. Yeah? So you have a finite volume, a reactor, and the influx to this reactor are short fragments, monomers, dimers, trimers, yeah, and they react, they bind to each other, they ligate, they do templation and so on. Yeah? So of course you get longer strands. And the interesting feature here is that the strand length distribution that emerges at high dilution rates is just a monotonically decreasing strand length distribution, which is close to exponential. We have a semi-log plot here. However, if the dilution rate here sort of is lowered, so that means that the strands have longer time to react, we get a peak. Yeah, we get a clear sort of non-monotonic behavior with a typical strand length that develops. Yeah? And this can be understood where it comes from. It's a non-equilibrium process where different timescales compete. But the position of this peak basically and the height of it depends on the outflux rate. So it's a physical parameter. It's not like a rate constant or something from chemistry, but it's a physical parameter that describes this. Okay, so let me come to an end here, I think. And in particular, thank the people. I mean, our hope as physicists, to summarize, is basically that physics could be like a catalysis that drives chemistry towards biology here. Yes, we have sort of a non-equilibrium enhancement. And let me thank the people on the left here who did the work that some of which I described, and of course my collaboration partners listed here on the right, and you for the attention. Thanks a lot.
All right, thank you, Ulrich. Questions? Any questions in the chat? No.
Since we are running into discussion, maybe we can have a little section of a discussion for answering questions as well.
Vlad does have a question.
Yes, thanks for an interesting talk. A question is about the what is the source of this equilibrium, the physical source of the disequilibrium, and what is the impact of the temperature on the network?
Okay. So maybe I can show this slide here. Yeah, so in these systems that the Dieter Braun studies, so there the physical source of disequilibrium is really a temperature gradient. Yeah, so he has these fluidic columns that are shown here, and there's a warm side on the left-hand side and a cold side on the right-hand side. And there's also gravitation. Yeah, so these are like vertically oriented or in some angle, and that means the warm water will sort of move up and the cold water will move down. That's sort of these convection circles that you get here. At the same time, charged molecules like RNA, they have the tendency to move. RNA in particular has the tendency to move from warm to cold. Yeah, so there's actually a directed motion of the molecules that we don't see here in the plot that go from left to right. Yeah? And the superposition of those types of motion, they sort of accumulate molecules in the bottom right, and the accumulation depends on the strength of this thermophoresis. And the thermophoresis becomes stronger for longer molecules. Yeah, so the thermophoretic Soré coefficient, that's sort of the name for it, that describes how strong this drift is that goes to the right, that becomes stronger as the molecules are longer. So that's one source of non-equilibrium. Another source of non-equilibrium is that you have an influx of small molecules and have an outflux of then larger molecules. Yeah, so you like a chemostat or something, you keep it out of equilibrium in a mass sense. And then in an energy sense, there's also a disequilibrium, because we always assume that in the background, there are chemical reactions that activate. Yeah, so whenever you don't get ligation, if you don't have pre-activated somehow in situ the monomers and the strands at the ends, such that the bond formation actually happens. So I would say there's multiple levels of disequilibrium.
Thank you.
Thank you, Vlad. So in the interest of time, I think we'll defer questions to the discussion, right? So yeah, so thank you very much to all the speakers for staying on time and doing such beautiful talks. And now I guess we will see you in the breakout sessions. Thank you.
If we have the discussion leaders here, then it would be great if you could introduce yourselves and what topic you would like to kind of focus on in your discussion.
Hi, can I go?
Yeah, Sudha, please.
Hi. Hi, everyone. My name is Sudha Rajamani. It's uber late out here, so before I completely doze off, I'd like to start with some discussion and then hand it over to somebody else who can probably continue leading it. Wonderful talks today. Thank you very much, Irene and Nita, for having me as part of this session. I'm very interested in understanding both the emergence of compositional heterogeneity and what are its implications for the various prebiotic reactions that we try and understand in the context of emergence of a protocell. This ranges from emergence of informational and catalytic molecules to compartmentalization and how components that are involved in your starting reaction mix actually would affect the output of the processes that you otherwise study under extremely laboratory buffered conditions. So very broadly, this is the stuff that I'll be touching upon in the discussion, in addition to being open to discussing some of the questions that came up during the talks too. Thank you.
Great. And I see Moran. Okay. Yes, we can hear you.
Hopefully.
Great.
Oh, that's great. Okay. So hi, everyone. Yeah, my name is Moran Frenkel-Pinter. I'm a new assistant professor in the Institute of Chemistry at the Hebrew University of Jerusalem. And I would like to focus my kind of discussion breakout room into kind of asking, what are we looking for when we talk about evolution of molecules? Is emergence important? Is continuity? How can we identify it, and what should we be looking for? So kind of looking at the process, and yeah, of course, in relation to everything that has been and all of these great talks that we had today.
Okay, just I see that Vlad has his hand up. Is that an old hand or is that a new hand?
Old hand.
Oh.
Old.
Well,
okay.
That's old, but I can use this opportunity actually. So let me lower my hand first and introduce myself. So not very many people know me probably. Yeah, my name is Vladimir Apitian. I'm a professor at American University and the senior astrophysicist at NASA Goddard Space Flight Center. So I study the history of the young sun, going all the way from the early Hadean to the late Hadean, and trying to understand all the sources of ionizing radiation that were available over the first few 100 million years, specifically first 100 million years, that was so important for the formation of the redox and also formation of the free radicals. So that's the question that probably wasn't emphasized during this conference, but yeah, I would love to see something, some discussion about it.
Okay, thanks. So we have one more discussion leader. Kristen, would you like to give your introduction?
Sure. So my name is Kristen Johnson-Finn, and I'm a new faculty member at Rensselaer Polytechnic Institute in Troy, New York. And my affiliation is in chemistry, but I'm also in earth and environmental sciences. So the subject matter that I want to cover in my discussion group is rather self-serving, which is let's think about how we can connect these different geologic environments to our experimental studies. And exactly what type of challenges are we encountering with that? I know that's a huge question, but I want to hear everybody's heated opinions on that and where we think we're going. So if you want to have that kind of brawl discussion, then join my discussion group.
All right, sounds good. It's good that we're doing Zoom, right? Okay, would our organizers like to add anything before we join breakout rooms?
I just wanted to say we made up the breakout rooms. We assigned the names that we got earlier. I don't know if they kind of map onto the topics that our discussion leads just covered. I'm happy to close out the rooms and rename them very quickly, just so that everyone knows which room each discussion leader will be in. Would that be of interest to you?
Yeah, maybe, you just can put our names out there because.
Right. Okay.
Yeah.
Let me do that really quickly before anyone gets in there.
Great. Thank you.
But as I do that, Jen, did you want to say anything, or did anyone else want to ring in as we kind of close out this first day with their thoughts or any other observations?
Yeah, I mean, just thank you all for such a wonderful day. That was really, really exciting. And just a reminder that as you are inspired by all these great talks and discussions, please we're happy to have you comment on the Google Doc. We would really love to get started. Maybe we can put that again in the chat, Zach, just so that if people want to jot some notes down as they think of them for things for the white paper, we'd love to have start to get outlined. Thank you all.
Jennifer and Zach, could you guys briefly tell us how to get to the breakout rooms? Sorry, I'm not sure I know how to do that.
Yeah,
I think I just opened them up. Did it pop up for everyone? I just renamed the rooms.
Yeah, it did.
Yes, there's a button at the bottom of the screen now.
Oh, I can see it now.
Oh yeah. Oh yeah. Okay, thank you.
Yeah.
Sorry.
Scroll all the way down. Scroll all the way down to see the rooms.
And for the close of the day, the discussion sessions have been set up. Again, we're going to end recording now for the big main session. The discussion sessions will not be recorded, so if it does turn into a brawl, as we've kind of alluded to here, as Kristen mentioned, that's just fine. This is meant to be, just like we do in a real workshop or conference. Everyone kind of splits off. They form groups to chat about what they saw today or what they want to talk about. So it won't be recorded and it won't be part of the official proceedings for the workshop. And hopefully it's just a way to try to unpack some of the ideas, maybe some of the new ideas that you came across today, and to kind of pick the brains of some of our speakers who have been gracious enough to stick around for the end of the day with us.
Zach and Jen, could you please paste the link for the Google thing, the Google Doc in the chat maybe?
I will do that. Yes, I'll do that right now.
Yes.
Well, thanks everybody.
Thank you.
Thanks everybody. See you
Yeah,
tomorrow.
thanks. Okay, let's go to our rooms.
Yeah.
Bye.
Bye-bye.